HD2 is good. We can stow away.
Copy. Okay. Crossing 50 meters altitude.
The bottom is in sight. See some rocks there. That's
Serial's
promising.
crossing 35
Rocks
meters
and
altitude.
sand.
Okay, coming to an easy stop on the winch. Auto heading engaged.
I sense a geological sample in our future.
Mm-hmm. This is like broken,
Have you in Sirius?
broken pavement. Slabby. My guess is they're
You have
a
joy
1
lock
meter
on.
wide, maybe. I'm not. We don't have our lasers on, so I can't really judge the size too well. But
Welcome.
they're about 1, maybe 1 to 2 meters, depending how far off the bottom we are. Pretty thin.
Weird about 10. Yeah, they do look like pancakes.
Mm-hmm.
We're at 10 meters now.
Copy. Spinning to port. Holding altitude.
Little fish or something over there in the top left.
And when the water column, I
Rotating
didn't
up.
see 'em.
Mm-hmm.
Pilots, the bearing to waypoint 2 is a 245 for Sirius.
Copy.
Look at this. What's that? A jelly of some
It
kind.
was a jelly. I was checking out the chat room. Silly me.
Okay, match your heading. Rotating up.
Here's something. Pyrosound, maybe? Nope. Tina-4?
I think that is, yeah, low bait Tina-4.
Sweet.
I'm at 4 or 5
Probably
degrees on the rotator.
Bathocyroe.
Ready? Paying out. Coming down at 10 meters per minute.
I think I see a sponge.
There goes another Bathocyroe. Oh yeah, I see that sponge out there.
That was a cool fish at 6 o'clock. What do you think this is at 3? It looks almost like line, but think that's human debris over there?
Difficult to tell, but yeah.
We're pretty
Bridge is ROV nav.
here too.
Is there more of it straight ahead?
Yeah.
Want to inform you that we are on the
We're
bottom.
pretty far
I would like
offshore.
to verify that the center rotation is set to the A-frame at this time, and when you're ready, we can discuss a brief bailout plan.
Sponge stalks.
Good copy. Thank you.
Could be sponge stocks.
Of course, that's an engineering video.
It's Roland, I thought they were like a couple feet long.
Copy. That's pretty close to high pack 245.
Seen some that have been about 3 feet long.
Oh, neat.
So it'd be nice
some
to
that
get
dead.
some
Okay,
of the
I'll-
rock
I'll
probably
rotate
from
to
the
port
Go
bottom
ahead,
as
Bridge.
you're heading.
and the top of this structure, I would think.
Copy
did
that.
someone
If we could set
call in?
bailout to east, due east, a 0
Hi, Stephanie. Scott, friends
9-0.
calling back in. Glad to see we're back on the bottom of the ocean.
Yes, too.
It's
Good
exciting.
copy. Thank you very much.
There could be some good rocks to grab here.
Copy.
Yeah, these look
paying
like
out.
slabs, right? So I'm not a geologist, so you're going to get a very elementary version of the habitat out of me. But these slabs are pretty large. They're like 1 to 2 meters wide, maybe, and they're fairly thin. So more like of a pavement, like
I'm
a
at
chunk
4
of
or 5
pavement,
degrees on the rotator still.
than necessarily like a rounded boulder.
Okay.
We got a nice
Over
white
here,
coral
20
at 3
meters
o'clock.
altitude.
Looks like it could be a gorgonian.
Copy.
Yep.
So the sand looks like it's fairly thick underneath these rock boulders. Right, like you could sink your hand into it.
Stick
Really,
your
so
hand through it.
Yeah, it's probably pretty soft.
We may see when we make a rock collection.
Yeah. Which is similar. That this is that thick sand we saw on the dive yesterday when we hit bottom.
Yeah,
There's that nice
nearby
thick
here.
sand. Yeah. There's also some yellow, I believe gorgonians or nidarians at 3 o'clock on these rock tops.
Seeing all these rock slabs is interesting, and it makes me wonder if we are going to be wrong about this being a giant coral mound, or if it's some sort of interesting geology.
Okay,
out here.
Ryan Stoge. should
I guess this
deploy the
isn't a coral
sonars?
mound just because I don't see any Lophelia rubble. And usually at the base of all these Lophelia mounds is always rubble that has, rolled down or
Copy.
whatever. The Lophelia mound's actually built out of that rubble mass.
Now,
so
are you ready for a Niskin?
I'd be very surprised if we have an actual
I'll check.
bioharm
I'll check with
Lophelia
Watch
reef.
Lee. I
I'd be
just
more
wanna
apt to
check
say
the
this
nav
is probably
first.
some kind of
Yeah.
rock feature.
So,
Yep, I agree. And we
Watch
saw that
Lead's
on the first
co-piloter.
dive, didn't we,
Are you
when
ready
we
for a
settled
Niskin
down,
sample?
Below that
yes, please.
bioharm.
All right.
Mm-hmm.
Resetting.
I think I see Stichopathes at 9 o'clock.
I see she's reporting the channel, so I think you're clear.
All right, and firing Niskin in 3, 2, 1.
Okay, that's water sample number 2.
Copy. Okay, resetting again. And firing in 3, 2, 1.
Did that one take?
I gotcha. Thanks.
It looks like maybe a yellow
So
sponge up here. It could be a yellow hexactinellid,
the Hertwiggia, maybe?
possibly. And then I also have a small coral at 3 o'clock, little white fan. Looks like it could have an ophiuroid on it. And then in the distance, okay, yeah, so that's at 3 o'clock. So if you swing right,
Copy, rotating up. Here, I'll turn off the aux.
number 3 fired.
Number 3 fired, yes, and number 2, it just stayed open.
Number 2 did not work. So we are, okay. So that means we only have 3 samples left.
2 samples.
2 samples left.
Middle and end of the benthic transection.
Right, because we did a 500
Mhm.
and then fired 2 but didn't work. And then number 3 is really this bottom sample.
Yes.
Then we have 2 left. Okay.
Copy.
Great. Now we get to look around.
Yeah, if you guys are ready to start looking. Okay.
Yeah, if you guys are ready to start looking. Okay. Yeah, if you guys are ready to
Nav.
start looking.
Nav, can we get the starboard rail?
Unfortunately, the starboard rail is, I can pull it up for a second, but it's finicky.
Okay.
Yeah.
All right.
I can leave it up for a bit, but it was going to be distracting to have on the whole dive.
Copy.
You can see it a little bit from here.
Yeah, that one went very quickly.
Fantastic.
But the upper is really slow.
Fantastic. Fantastic.
All right, co-pilot, when you're done with rail cam, I'll turn it back off.
Copy. Yeah, you can go ahead and do that now.
It might be
Okay.
someone shoreside with their phone on. Is that again?
Possibly.
If you're shoreside and have called in, please mute your phones. Thank you. Yeah, there's a Carl dead center right in between your lasers. If you could just zoom in on that guy.
Oh, we got the lasers on,
Copy
so
that.
we have a better feel for
15
the
meters,
size of these
250.
slabs. This one's definitely more than a meter. And we're going to get a look at our first
Yep.
benthic organism here. So we have a nice,
Co-pilot, I'm going to bump you in 15 meters.
that bamboo
250 is the
coral?
direction
Maybe.
of ship move.
And I would call
Copy
that,
that.
astroschemia,
Bridge is
but
RV
I'm
nav.
sure. Oh, and a Faria. Look.
I'd like to call on a ship move if you're
yeah,
ready.
you see the glass sponge.
Range
Faria,
15
Faria
meters,
Aka,
bearing
probably.
250 degrees, speed decimal 2 knots, please.
This is looking like a primnoidae
Primnoid.
octocoral to me, not a bamboo coral.
Okay.
It's a very common deep sea family. And I'm even seeing
Good
something
copy.
way
Thank you,
behind
Buck.
it that is unbranched.
Oh yeah,
Tell
that
from
yellow.
here if that's,
So I'm at 260 right now.
I don't
Pilots
see it
have to
yellow,
rotate
but
10
maybe
degrees
well, you
to
definitely have a
port.
better view than me. Yeah, that must be a
Okay.
psychopathy, as I said earlier. But yeah, this looks like a primnoid. You can see that the polyps are arising in these whorls. I can't tell right now if it's 2 or 3 polyps per whorl. They're sort of connected near their bases. Very classic.
It looks
sort of
like
pattern.
3. I see 3 per.
Per node.
Yeah.
Yeah.
Yeah,
It'll be-
so not sure if this is genus Paracalyptophora or Calyptophora. Have to look into that a little bit more. I'm constantly forgetting my primnoid genera. There's a lot of them. That's great. We have a black coral and an octocoral next to one another.
Yeah. Yeah,
Nice.
it'd be great to take a look at that.
Great, and pan down
Sponge.
to the associate.
And this is my guess is astroschemia, but I'm not an expert in these. Long spaghetti star. It's a type of ophiuroid or brittle star. They're usually associated with these gorgonians. They like to crawl up off the ground and hang out in the gorgonian so they can feed in the water column. If I am telling you any wrong information, I'm sure Chris Ma will chime in and yell at me. So I will be corrected shortly.
No worries. I think there was
Oh,
a
the full
whip
specimen?
behind this as well. That'd be worth taking a look at.
If you guys can look up at around 2 o'clock, there is a nice white, wrinkly sponge. Yep, top corner.
Video's clear.
There might be a shrimp here underneath that Stichopathes. Yep. Ooh, look, maybe a corallimorph or a solitary coral here.
Right on the top of that rock
Yeah,
to the
right
right.
at 3. Squat lobster. Oh yeah, squat lobster. Look at that.
Yeah. Oh, we got lots to look at.
We got plenty of time, so.
As usual.
Means it's a short distance dive. Like we don't have a lot of area to cover, so we can, chill and zoom in a bunch of stuff.
Zooming in on this glass sponge. This is almost certainly the genus Pharea. And what's that just to the left there? Is that a small urchin?
Yeah, it looks like it to me.
I like the looks of that urchin.
Yeah, he's cool.
John Reed is commenting in the chat that it's possible that this is an allied
Go ahead,
group,
Rich.
Euretidae.
Good
thicker.
copy. Thank
But
you.
it is a bit thicker than a lot
Pilot's
of the Phareas.
last move is complete.
I think
Copy.
he might be right on that. Be good. Can we zoom in on that urchin a little bit? I think we'd like to get some imagery of that.
That was max zoom.
There's actually quite a bit of stuff on this rock when we zoom in.
Science, that was max zoom.
Yeah, he's tiny.
Are
Less
tho-
than a centimeter.
Oh, wow.
This tiny little thing. There's also a white-tipped. I would call this a solitary coral, is my guess on the left. I'm sorry, the right at 3 o'clock. But I can't really see its helices, so I'm not really sure.
Rather than a corallimorph?
Yeah,
maybe?
I think so, but I'm not 100% on that. I guess I'm not 100% on anything I see down
Well,
here.
I understand. Same. But so corallimorphs are a lot like they're the sister group of the scleractinian corals, and their main difference is that they don't have a calcium carbonate skeleton that they're. So we
That
should
could
be able
be
to
a-
tell.
That's cool.
That is really cool. Now, is there a skeleton in there?
I don't see one, but it's like a clear anemone. Bubble tips.
It's
Yeah.
It's quite.
It's very
There's
squishy.
some slack
It's
in the
squishy. It's
space.
beautiful. But I'm curious what you can see the color inside. It's not a red pigmented gut, is it?
It looks like it here.
Yeah.
It looks like this ring on the outside, though, is probably not calcium carbonate. That's probably the rock behind it.
Right. So this probably is a corallimorph and not a solitary coral.
It looks like a green sponge here at 9.
Yeah, you can see the oscules, the excurrent openings of that green sponge, just to the left of this beautiful corallimorph.
Yep. I was tracking to the sponge on full screen.
It's fantastic. And there's another sponge just to the top left of the green sponge as well. Another demo sponge, a white one.
Video's clear.
Did we do an on-bottom Niskin? Yes.
Yeah, we did
Okay.
that.
An orange in the sand
So
directly
our
above
next
that
collection will
yet.
be our third.
And a black coral to the left edge of the screen.
Looks like there's some cyanobacteria, or there was a squat lobster, but I think he's disappeared. Yeah.
Now the small yellow octocoral, is that the Gorgonian and
At 9.
maybe Paramuricea or something like that?
I'm going to
Could
rotate
be.
down.
We could take a look.
Yep. There's a. Yep. He's flopping around in the breeze right there. He's right at noon now.
4-5.
Yeah.
Right above laser.
Yeah, that's a black coral.
Oh, that's a black coral.
Probably
Thanks.
the genus Storopastis. I think you'll see when you get closer, the polyps look quite a bit different from.
Yes, they will.
that.
What genus did you say? Oh, yeah.
Stauropathes, we've already entered it into the annotations
thank you.
in the chat room.
Thank you. Oh, nice. So black corals are really octocorals.
Flight
They have
tilt
8
down.
tentacles, but they have a
No, they're not
They're not
No, they're
octocorals.
hexacorals.
Hexacorals.
Yeah, they have
Hexacorals.
six.
Six
One of
tentacles.
the cool things, this is a great view, actually. Each polyp has 6 tentacles, and what you can see is the polyp is all stretched out along a branch. It looks like the tentacles are coming out in pairs, and where you see that little red tube sticking up in the center,
Yeah,
that's
ship
the
move
mouth
completed
leading into
about
that
5
weird
minutes ago.
stretched out polyp. So it almost looks like polyps with 2 tentacles, but they're actually 6 stretched out.
6 tentacles.
Yeah, that is a great view of that. A lot of times it's a lot less clear.
In fact, I always tell my students it's really bizarre. I believe black
Yeah,
corals are the
Maxine.
only hexacoral where the polyp actually has 6 tentacles. All the others, there are always multiples of that.
Okay, video's clear.
So true hexacorals.
And then in front
And
of
then
it,
the whip is another.
Yeah, likely
Pull into the
Stichopathes.
stick.
The black corals, yeah. It's yellow. I don't think it's Leucini. Leucini is the one we have in the I would say shallow water, but mesophotic. This one's yellow.
Yeah, and you can see how that's a little harder to see the polyps with 6 tentacles.
Looks like 2.
Mouths are more difficult to make out because the polyp sizes are so much smaller.
Yeah, let's look at the base.
What do you got there? A white thing on the rock. There it is.
That's a small. It's an octocoral as
Yeah,
well.
okay.
You can see the.
Polyp.
Just to
Weird
9
little,
o'clock.
barnacle?
Where are you looking? What?
It's got too long a stalk for a barnacle. It must be a recruit.
Oh, at 9 off. Now it's off screen. It looked kind of like a Gorgonian,
Yeah.
maybe.
Oh,
Yeah, the little white one that we were talking about.
Maybe like
Look
polyp
left.
recruit or something.
Yeah, it looks like it could be a recruit. There we go.
gonna zoom in there.
That's max zoom.
Yes.
Oh, I see. It's actually got 3 or 4 polyps, doesn't it?
Yeah.
Yep.
4. 1, 2.
Well, that's a good sign. It's telling us that there's recruitment here. It's
Yeah.
not just all old corals from long ago.
Video's clear.
It looks like there's a little brown guy back there, maybe a clam or something. Okay, we're happy whenever you guys want to move on. We're taking our grand old time, though, because we've got a short transect today. So, lots of
Yeah,
zooming
they
and
were
stuff.
interested in rocks.
All that.
And I think we'd like to collect a rock, so if you find one that you guys feel like you could pick up real easy at the claw and will work for you, we're happy to take really any one you want. I don't think so. Just
Hey, Stephanie.
yeah. Hey, John, what's up?
Yeah, when you're looking at the rock close up, it looks like the typical deep water phosphorated coated limestone.
Yeah.
And it's pretty common out here. I don't know if there's any geologists on board.
Yeah,
but,
I think so.
Not on board,
like
but
that
maybe on the
phosphorus
chat.
coated.
I haven't
If-
seen it.
if you
someone
kinda
in the
set
chat
up on the,
necessarily yet.
Yeah, so like the
Maybe the
Miami
right side of
Terrace
that
is
rock,
made of
you might
this
be able to
block,
reach
like
up.
plaque.
Actually, those look
you
pretty
know,
thick as well.
I guess it's phosphate.
I think that one down there, the
right?
UV laser's right
This
over
is-
right
yeah,
now.
it's a common.
Yep.
It's the common rocks. This is interesting though. There's like a shaley shape.
Mm-hmm. Looks like more like consolidated of sedimentary form.
Hydraulics are spun up.
So I think we're lining up to do a rock collection. It's too bad that corallomorph didn't look easier to
Yeah.
to pick up. Scott was commenting that he thinks that of all the Cnidarian groups, they're the
Rotating
least
down a
well
little bit.
collected in the deep.
Yeah, the little bubble tips.
In fact, Alan, I can't think of a single collection we've made of a corallimorph.
Well, we can go back and get it, or try to get it.
Well,
might-
I think it needs to be on a substrate that's easily picked up. But otherwise, you're going to have to scrape and claw, and who knows what kind
Right.
of shape will come.
So if we
Not
find
every-
one on a small rock, it might be worth it.
Yeah.
Exactly.
Yeah. This one looks like it might have a little lollipop sponge on it.
Oh, a little clatorizid, maybe?
Maybe. And those are the carnivorous
Yes,
sponges.
those would be carnivorous sponges. So unlike most sponges, well, let's see when we get a little zoom in here. But if it is a carnivorous sponge, that would be pretty exciting.
This is going to be sample number 3.
$3G, yeah?
Want me to put the mini Zeus up, pilot?
All right, so the sediment's really fine. You can see it just clouds up when we touch it. That works for us.
Nice. Well,
Do you want me to bring it
Sponges
inboard,
on it?
pilot? or-
definitely some sponges.
Okay, ready?
Little ball
Alright, coming
sponge.
inboard.
Reminiscent of Geo today, but
Oh, geez. I can't.
who knows?
Yeah.
right?
Well, we'll find out when we bring it up.
That's not fully inboard yet.
Look at under scope.
And I
if
can-
you
Right, I
want.
was
Can't
watching
tell
the
those-
temp probe.
Look like maybe dead hydroids blowing in the breeze
okay.
there. They've definitely
Alright.
got a lot of sediment
Alright, that's
collected
fully
on
inboard.
them. On the other hand, it's possible they could be alive, and we will examine those when we get them back to the ship and preserve them if they look like they're alive.
Maybe tiny little corals on this rock. Some solitaries or gorgonians or something. All right, so it's about a 10 centimeter rock.
Copy, coming up.
That's going to be great to look at.
It's like a cobble piece, cobble size.
Stop and rotate. Bump up a little bit,
There's lots of stuff growing on it.
a little bit more, and stop.
Yeah, looks like solitary coral there. Dead center, right?
Yeah, it could be. Some neat little sponges.
Yeah, got a lot of little things growing on there. Lots of sponges.
Maybe some worm
I think
tubes.
you guys are going to be up late tonight because there's an awful lot on there. I think a black coral as well, maybe.
I told Mr. Collect It over here that he's not allowed to fill every bucket. So,
Well, we will.
Alan, I disagree with.
You agree with me?
with you. But I'm just saying, yeah, this rock alone has got all kinds of wonderful stuff on it.
Yeah.
Do they want to put it in a rock box or?
No, go for it. Rock box is good. You doing port side?
Looks like a success.
Wing outboard.
No, that's the first rock sample.
Looks like it didn't. Okay, that's full in.
Well done. Great collection.
We have a pretty cool fan at 3 too. Not sure if that's a coral or a sponge. If you guys want to move there when you're done, when your arm is stowed and you're comfortable to get moving.
Okay, drawer coming out. Oh, I thought I was proud.
That looks like Stickopathes to me in the center. This orange whip coral. I'd have to get a better look, zoom in, and check its polyps out because we also have like an Eli Zelid that looks similar to that. It's whip-like.
Yeah, I agree with you, Stephanie. That's going to be a Stickopathes.
Yeah.
Something in the genus Stickopathes or closely related to it, black coral.
But this is separate from the other whip that we looked at.
Yeah, I think the other one was a yellowy color, much smaller. We get them in gray up in shallower water. Go ahead.
Yeah, there are currently, I believe, two whip genera, Stickopathes and Cirripathes. But some of the genetic work has shown that there are actually two pretty distantly related Stickopathes groups. And so I think this orange one belongs to the other one that may be closer to the family Aphanopathidae. I don't remember exactly, but it's still called Stickopathes. But for sure, we have two different species right here that we're looking at, the yellow one.
The yellow and orange, yeah.
We have that same problem with hydroids that
have it with
branch.
everything. Have it with everything. Yeah, the white fan at 3.
There it goes.
It's just right at the edge of the screen there.
Bathocyroe floating by.
Nice.
Looking like food to some of these filter feeders, maybe.
Just from a distance, my guess this is going to be a type of Phycalia or elephant ear sponge, but we'd have to get closer for me to really confirm that.
HPU secure, valve packs disabled.
Fish in the top left there, eel.
Yeah.
Copy.
So that says something brown here and coming into the center screen around 4.
Just below the fan.
5.2
Not sure what that is.
on my rotator.
Yeah, when you look close at these rocks, there's a lot on them. Come in for zoom here. Nice.
So if we look on the other side of this, it might have that veining.
The elephant ear veining.
Yeah, I don't know if you'll be able to get to look at the other side. It might be in an awkward place for you guys, but yeah, only if you want, can manage. Otherwise, if it doesn't have that veining, I'd probably call it something similar to Pachystrelid.
Copy.
If it does have the veining, I call it Ficalia.
Maybe Christina will call
I
in.
think port
Yeah, I
side
don't
will
know
be
if she's
clear,
watching
starboard
today.
side,
Recognizes this.
which I don't really have
Oh, it
eyes
looks
on.
like garbage down there too, huh?
Oh, copy, copy.
Plastic.
I'm going to rotate a little
Very
bit to
thin,
the
that
right.
fan.
It,
Can you come in a little bit on Sirius video?
Yeah, that plastic bag is awful.
Thank you, video.
that sponge.
This Stephanie.
Yeah, John.
Christine is on a Zoom for until about quarter after, I believe. So we'll give my
could stow that
expertise
swing
of
arm.
spongeology. When you zoomed in on it, it looked very, very porous, like a hexactinellid. But from a distance, it did look like a pacastrellid. You know, that
Pull the starboard lower in and maybe get a little bit of light underneath.
Yeah, and we're
that.
looking at the opposite side of it now, John, and I don't really see that veining. That usually that veining
What
is
if we
really
tried the extra
like
light? I think that
a
would put
vein,
it there.
you know, like really distinct veining.
Yeah,
So you're thinking Pacastrella,
Okay.
John?
no, he was wondering if it might be a glass.
Oh, a
sponge.
glass sponge.
Yeah, I mean, this
Okay,
view, it looks like
fog's coming up.
pacastrellid.
Oh,
That's
okay.
10%
Other
on the
side,
fogs.
it looked very, very
Did not
porous,
see a
like a
change.
hex. So
Okay.
I thought it got a little bit lighter.
how it's kind of fringed around the edge, I'll vote on
Okay, that's
pacastrellid.
20, 25.
And there's that stupid plastic bag.
Adjusting the sediment
Yeah.
filters.
Yeah, we're gonna call this Pacastrella.
It's more than I've got.
Maybe-
Okay, I'm going to swing the starboard lower in.
Yeah, I think we can
Yeah, we're
pull
good whenever
back
you
and look
guys
at
are ready.
something
I
else.
just I don't have eyes on it, so
Full screen.
Now, do you want to see the brown organism that was a little bit closer to the ROV from
Yeah,
here?
if you want to, well, I'm happy to
Show
do
it
that.
without the bag, but that ain't going to happen.
Sort of bushy about one rock
Video's clear.
back. Let's see, maybe to the left of here. Or we can keep moving our way up. I'm sure we'll come across, well, shouldn't be sure that we'll come across that exact species again, but we can hope for it.
There's also this blue. You were saying you thought that was my Cali?
No, I think, I don't know, but there are shallow water blue sponges in the genus Halasarca.
Halasarca.
copy.
We have another Gorgonian. It looks like it at noon. It's been flopped
Fogs
over.
are out.
Yeah, more Sticopathes.
Mm-hmm.
And the yellow, that was Storopathes, was that correct? The bush-like black coral? I think we just went out of view.
Yeah, that's right, Alan. Storopathy's kind of like a little fan.
Yeah, thanks. I think that, yeah. Wait a second.
Not this one.
No,
This
that's
is a
not. That's
Paramaricia,
an
or at least in
octocoral.
the family Paramaricia.
Paramurciid.
That's no longer.
Uh-huh.
Yeah.
Great.
Just
Actually,
looking
now
right
the family
behind it.
Plexoridae, but something like Paramaricia for sure.
Mhm.
So this is an octocoral. This one.
So that's new.
That white thing on top of the rock there looks almost like a stylaster knocked over.
Yeah, it does, doesn't it? And I also think I see a stalk crinoid at 9 on the rock that's right below us. There's also a fish down there. You can see his black tail.
Can you zoom in on that coral that we just passed on the right?
Yeah, we can come back to it. Since we just blew past it, I will come back to it, John. Yeah, can we look at this? It's at 1 o'clock. That
It's really nice stock coral.
Crinoid.
Yes. Did I say stock coral?
Yeah.
Stock crinoid. Looks like it has an associate on its stock there.
Yeah. Oh, neat. So this is a stock crinoid.
What is going on?
Looks like a hermit.
Is that a hermit?
Mm-hmm.
Oh, I see. He's holding on. It's got a shell.
That's a great view. Hey, before you pick up and move away from this after you've got this, just look off to the left on the next rock. I don't know if it's another one of these stock crinoids, but there's something odd. I almost thought it was like a Metallogorgia or Chrysogorgia colony, but
Okay.
too far away to tell. Thanks.
Yep. Cool.
You don't usually see the hermit crabs up on these crinoids. This is a bit of an unusual observation.
Now, as we go back and get a better view of these rocks, we've got a comment on the geology here that these look like sedimentary rocks which have been exposed along the continental slope, and the black color is ferromanganese crust created by minerals that are
Showing
deposited
two
from the
good-looking
seawater
corals
on the
upper left
surface
and
of the
up
sedimentary
lower right.
rock.
Awesome.
The crust grows extremely slowly, so these rocks have been exposed for quite a long time.
Hey, pilots, can you go back to that flattened stylaster white fan coral that was on the rock before? It should be to your right.
Hey, wait. Before you go, Stephanie, all you have to do is zoom just to the left of where you were looking.
Zoom to the left. Okay, wait.
You don't have to come
Before
back.
you run off, there is a 9 o'clock
Hold on,
coral
pilot.
real quick in this shot. Sorry, I'm getting bossy, guys. Okay. I get to get really pushy back here. It's real easy to tell them what to do. Yeah, 9 o'clock, it's a stalk on the rock right next to the rock we were just looking at. So it's just about to come into center. Yeah. And what do you think this is, Scott?
Well, I wasn't sure. It could have been a Metallogorgia or Chrysogorgia skeleton or just another whip with hermit crabs or something on it. That's why I wanted to have a look.
Yep, dead center. There you go.
I was hoping we'd move that we could
Go
have
to the
zoomed.
top of it.
It looks to me like a dead thing with a Hermie.
Okay, so it's the same sort of thing. We've got a hermit crab again crawling up a stalk. Maybe it was a crinoid stalk, but this is interesting seeing two of these next to each other. I haven't seen that before, and then all kinds of hydroids.
Oh, it's
All right,
so
that's
many
great.
hydroids. I like it.
Okay, I think we're good, pilots. If you can
Yes,
go
clear.
full wide, and then there's a toppled over stylaster we want to get a quick look at to the right. It looks like a white lace coral. Yeah, ahead there's something crazy going on too. Yeah, there's that. That one's good. That one that's standing up. But John was in particular looking at the one that was toppled. It looks like another faria there, maybe.
Another glass sponge.
Oh, you hit it. Yeah.
If it's a euretid, might be.
He should be on this rock right here, I think. Or near it. There it is, right coming up into screen, yeah. Ooh, I see a sea star. Right there.
All right.
Yeah, that's good if you could snap zoom for us.
Calling Dr. Ma.
That's a pretty big one if it's stylaster. I think it's stylaster.
It has that.
Oh, no, it looks like it might
No?
have polyps. Yeah, it looks like a toppled gorgonian.
Yep.
So that's interesting. Still alive.
Little shrimp hiding under
Yeah,
it,
a little something behind there.
I think.
Shrimp
Yeah,
or
this
fish.
looks to me more like a corallium or hemicorallium, something like that.
Corallium.
Oh, definitely an octocoral. One of the precious octocorals.
Let's look at the base.
There's a little hermit under there as well, tucked away. Now, where was that star you saw?
The star is below it, like more
The
towards
full
the
screen?
the 6 or 5 o'clock time when we're zoomed full wide.
So
And we
full screen.
do, I can confirm that Chris Ma is in the chat room.
Okay.
And if
There's
he's,
another shrimpy.
yeah,
Shift over to the star.
Yeah, there's a star, a little white star. There he is. He's just
very thin
dead
arms.
right of laser. There's a neat, looks probably a hexactinellid below.
Oh gosh, no,
Really thin. I just
Was
call
it
those
wider?
ice spongage,
Yeah.
because they look sponges, because they just look like a layer of like crusty ice to me. I don't know what they
Partial.
are. They're not they're not uncommon though. Those little thin crusted white hexactinellids.
was it sticking up or encrusting?
I think it's encrusted like
Yeah,
this,
that's-
like flat to the.
pretty unusual for glass sponge.
It might not be glass sponges. It might be a demo. This real
So
thin.
It's be interesting to look at this star. Chris comments that it's appears to be it's sort of hunched over, like it may be feeding.
Yeah. That's neat.
Can't see what it's feeding on.
Rock schmutz, I guess.
Maybe.
Nice. Did he have a name for it?
No name is forthcoming so far from the chat. But oh, Henricia.
Oh, okay.
Sorry.
Yeah, the brownish discs. I don't know what those are.
Would be nice if we can. I'm not sure we can get any closer, right?
That's next soon.
Yeah.
Yeah.
Yeah, so if this is Henricia, Chris reports that it's most likely eating a sponge,
Okay.
of
Maybe
which
it's
there
good.
appear to be many around.
Yeah, I think that's probably what these little, like,
Yeah,
dime-sized things are.
hmm
Little white sponges, little brown sponges.
Thank you, Chris.
All right, we're happy whenever you guys are. It looked like ahead of us there might be a really cool, like above, you'll have to like pick your head up, but yeah, far in the distance there might be like a long stalk and like a fanny thing on the end of it, so. The fanny thing. Yeah, coral fanny thing. There's also maybe a cutthroat eel. This is interesting. Single stalk with like a top with like a bush. I'm not sure what it is.
Yeah, I feel like I've seen a picture of that somewhere, so hopefully Scott will chime in and let us know.
Almost looks like a hydroid on the end of a Sticopathes, but I don't have a clue. That's odd. There's another one behind it. It has to grow like this, whatever it is. Ooh, and way up there at noon. And then way back up top.
Big fan in there.
Yeah, if we get a zoom in on this guy and see if we can get an ID from shoreside. Oh, there's definitely a Gorgonian. Oh, weirdo Rama.
So great. Another one of those brittle stars in it.
Yep, sorry guys, I'm on a separate call as well. So this is Metallogorgia classic octocoral, and it's always found with one brittle star. We know from around the world where this has been collected that one species of brittle star lives only on this colony, on this species of coral, and the species of coral only ever has this one brittle star associated with it.
And do they always have one?
Association. And this is on the shallow end of the range for Metallogorgia melanotrichos, so it's kind of interesting to see it here. It's clearly doing really well.
Some of those polyps look distended.
So I think there are two of those, right? We saw one or something that looks quite similar behind.
Mm-hmm.
Some of those polyps look distended.
This is a really nice close-up, and there's some kind of funky branch tips there. Those curled ones that you see on the right side, look like they're extensions of branches. That's kind of odd. You notice everywhere else it's got a very regular sort of bifurcating growth pattern. Gives it this very geometrical shape. So I'm not sure how to explain those wiggly curled ones.
And they're also lacking
I've never seen that before.
lacking polyps. Or are the polyps retracted?
I mean, they're covered in tissue. They're alive, and then, yeah, you can see some polyps at the very tips of them. That's weird. That's really weird.
That's what I like to hear.
Okay. Yeah, me too. Thanks for finding that.
Hey, Scott, can you type that into the chat?
I've got it.
Just so I can spell it.
If we can get the, all the way down to the clip.
I think I already did.
You did? Okay. Thank you.
Right after my comment of, ooh.
Yeah. And then, yeah, there's another one behind there.
So this we think is maybe in the shallower end of the other depth range. Ooh, there's some, I guess, a cutthroat. Yeah, there's another one here.
Yeah, Stephanie, I don't know if I misheard you there. Normally they're deeper, so this is the shallow end
You
of the
take a
range.
snap at one of those joints in the thing there. It's almost center.
So that's looking at its stem. It's very textured.
Yeah. It's an interesting bend in it.
Video's clear. Science is clear.
This is just...
There's some crazy stuff in here. Okay.
Cool.
I have my eyes focused on Noon in the distance. It looks like a really tall fan. Okay. We can make our way that direction.
2.7. The target waypoint is 2.45.
I mean, those elbow shrimps we saw yesterday.
So just today, yeah.
Those really sharp elbows in there.
Or 20.
Antenna.
Copy. 20 at 270. I'll call it in.
I'm at 4.5 right now. I'll rotate up a little bit.
Bridge, this is RVNav. We're going to call it a move if you're ready. Range 20 meters, bearing 270 degrees, speed decimal 2 knots, please.
Oh, nice.
Oh, let's look at that sponge. I want to collect it.
Of course you do. Is it
a stocked glass sponge?
It's not Hylanema, though.
No, I don't think it's Hylanema.
Hylanema has the really long stalk, but they're like this broken up, like pieces of fiber optic cable. It almost looks like. This one is more stock and stuck to a rock.
Oh,
and it's got critters in it.
Oh, that would be great.
You want to get this guy?
Yeah, is that collectible? That would be terrific.
Ooh, easy stop.
I think the stock itself is going to be hard, but the top looks like it's going to be fairly
neat.
soft.
Okay, so we're going to be wearing gloves when we get this out of the, for
Yes.
sure, because those spicules are going to go right through your fingers. So to be very careful when we take this out of the bucket.
Oh, yeah.
Sometimes they go right through your gloves.
Right.
Do you have a name for it?
I am afraid that I do not.
Okay. Hexactinella is good?
Well, it could
Believe so.
be one of these. Yeah, hexactinella. There's a couple different families that have stalks. Even within the Euplectellidae, that some
Can
are
you spin up
stalked.
the HPU?
But
anytime?
the taxonomy to figure these things out is in the details
full
of the
screen of the head.
fine spicules that are in the organisms, and
Bridge, this is RVNF.
Full screen of everything.
so
I'd like
We're
to
on die
request an
5.
easy stop at
is
this
gonna
time,
be
please.
Sample number 4.
yeah,
Thank
this
you
is
much.
sample 4B.
Pilot ship's coming to an easy stop.
I
You
would
got
direct
about 5
them,
meters,
let
so
them
you should
know
help get
yours
if
just
they
a
need
little
to
bit
get the
closer.
stalk if that's what you want.
It would be good to grab it by the stalk if possible.
Okay.
Gotta have
And then
another
it's going to be
measurement on it.
probably pretty crunchy.
The stalk itself should be very hard. The top, I expect it to be
Delicate.
kind of soft. Yeah.
Okay.
Oh, it's bigger than I thought.
Yeah, so it's about 15 centimeters on the top.
Video's full wide.
Yeah.
It has a huge oscule too, that big hole in the top. That's the X-current siphon where the water comes out.
Yeah, that's the X current.
Yeah.
Yeah. So the animal is drawing water in through the
Go ahead,
sides of
Bridge.
its body.
Good copy, I'll stop.
And
Thank you.
that stalk looks like it's almost fleshy.
Pilot's ship is
Yeah, it's
stopped.
made up
Co-pilot, you may
of
see a little bit of
parallel
swing.
giant
Copy that now.
single axis spicules altogether.
Rotate
There are
it
some
down.
that grow just a single
Mm-hmm.
giant
4-5.
spicule upon which, but that's not what we're looking at here.
This is
this is made up of multiple parallel spicules.
So if we were to cut it, you'd
Pilot,
see all
while
of
you're
them kind
setting
of
up,
like,
you have
like
all
fiber
bio
optic
boxes
cable
available to you.
Exactly.
growing in that, like almost like a rubber. It looks almost like a rubber hose, right? I don't know how to explain it. I know, like
Well, they
it's
have
like
this.
soft on the outside and then real sharp spicules inside.
They're actually secreted intracellular, so they're inside the cells. The cells surround them, and they secrete it on a non-mineralized core, and so that creates a little small
They are.
space, and then it's laid down on top of that layer after layer.
Neat.
So that's what gives it the
Yeah, I'm wondering, should I
property fiber
stow
optic
the
quality.
lower, starboard lower, if you slip
We thinking
off the
undescribed
toe?
species or just a
don't know,
target
it's probably
interest?
okay.
I would be surprised if this is described, but
Okay.
but a lot of the deep hexactinellid species that we know of, and there's 500 or 600 described ones, they have been described based on a single sample.
Oh, geez.
So they really are an under. Well, you can imagine, since they're found mostly in the deep sea, they're very undersampled.
Plus, and those samples probably came up on nets and net
Yes.
trawls. In which case, if this is coming up on a net trawl, I mean, it's getting destroyed.
Yeah.
This is like a fragile. Most hexactinellas are super fragile. So pulling that up on a net is just forget it.
There are
You know?
exceptions to that, but
Yeah.
yep.
Do you think they'll need to clip that?
I think it'll break
I
off,
think it'll break off. Okay.
but I'm new to this.
I mean, usually, if you've touched them in the lab or have handled them or something similar texture, you can kind of tell just from looking at them what they're gonna look like. Now, not always, right? Sometimes
Right.
I think a sponge is gonna be rock hard. We use the manipulator and touch it as a rubbery thing, you know. So
This is going to be like, it's like cotton candy.
got candy on a stick. Yep,
Zoom on
that
it.
should work. Agreed.
It'll definitely be like cotton candy
Yeah.
to some lucky researcher who gets to study this sample. This sample is going to go back to the Smithsonian National Museum of Natural History, and there it will be available for study by scientists around the world.
Mm-hmm. And we're going to take a DNA.
Yeah, we'll
for it,
definitely
too,
take a
actually.
DNA sample, and we will either sequence individual genetic markers out of it, or perhaps even work on sequencing the
Nice.
entire genome. That is great.
Awesome.
Oh, it's so soft.
It is very soft.
Yeah, it's even softer than I thought. But this is.
I need to squish it a little bit. When it gets here, if I want to take my gloved hand and just give it a good squish. Yeah,
It's
I'm probably
really-
going to get spicules in my
Yeah.
fingers. I don't want to do that.
This-
That hurts.
Overall growth form is going to be,
Okay.
partly important for the taxonomic description, but then also these spicules that are on the outer surface of it, and then in hexactinella taxonomy, they look at the spicules on the outer surface, on the inner surface, and then between the surfaces, and they're almost always different one from the next.
And they can also grow in different directions, too, can't they? And aren't
Cover that target
hexactinellids named that because they have six-sided, star-shaped...
Yeah, that's sort of like
Spicules?
a jack, if you remember that. Yeah, so it's like
Oh, like jacks,
Yeah.
like the 1950s
Yeah.
toys. Yeah.
390-degree
Yeah.
axes, And they some of the very smallest, they have obviously some very large spicules that are making up the base, the stalk, and then other spicules which can be they're microscopic, extremely tiny, and they make all these beautiful like star shapes, and even with really interesting little bumps and widgets on the end and stuff, and whether they
Little
attack.
barbells, like little barbells.
Some of them have barbells.
They're really neat.
Exactly.
They look like little pieces of, almost like someone blew a little tiny piece of glass,
Yeah.
like blown glass under your microscope.
So what I would guess is that this is in a group where like Bolosoma, but it really is a guess at this point.
And this will go 95, they wouldn't do formalin for this guy.
No, we don't need
He's gonna be
formalin.
95.
Yeah.
Okay.
Do you want to put it in the
And where are
port or
they headed with
starboard
this?
bio?
Port side. Okay.
Oh, open both boxes. Okay.
Port.
And extending both pins and drawing out.
That's number 4B.
I think that's going in the
full extension.
outside.
Port outer. Okay. Port outer's number 4.
It's just such a lovely animal.
I was watching, what was I watching? I think it was maybe it was like a Smithsonian show or a PBS show, and they took the sponge and they just kind of like squished it a little bit so that all the cells kind of came out, and then they watched it under a microscope and they started to reform. And I was like, oh my god,
Yeah.
it's amazing.
Sponges have some amazing cellular biology. It's true. And this group of sponges, the glass sponges, one of the interesting things about their cellular structure is that a lot of their bodies are made up of giant multinucleate, what we call a syncytium. So rather than like a single cell with a single nucleus, it's sort of a massive stretched out cell with many nuclei all through there.
Yeah.
And they have cytoplasmic streaming, which is something I think that we see in plants and stuff like that. So
Yeah, so Caulerpa, which is the shallow water algae, is the same, right? Where it's one single cell, but it can grow like hundreds of feet long, and it has, of course, millions of nucleus. It's awesome. Will it grow back from that stem?
It's a little
It will
close forward.
likely not grow back from that
Okay.
stem.
But if you were to rip the top off and let that float, that might grow back.
I suppose
It could settle
that's
down and...
possible. Yeah, I'm really not sure. how good the regenerative abilities are
Okay.
of from that
Pins are
level of
retracted.
damage.
Oh yeah,
They
because we
can-
took the whole thing.
Yeah.
Retracting drawer.
Success.
Hey, Stephanie.
Hey, John.
It's fully retracted.
Hey, I've been on
Nice
a
job.
faculty Zoom meeting trying to watch group at once.
Oh,
Not
sorry,
sure
pilot.
anybody has figured out what that was.
Do you know what it
I'm
is?
looking through our I'm looking through our deep sea coral photo album that we've made, Christina made, and it looks very much like rhabdopectella tinnitus.
Abdopectella?
Rhabdopectella tinnitus. Yeah, has a big head on it, a very thin stalk, kind of warty,
Oh, neat.
round the edge. So I'll type it in, but
Yes.
you can check.
Thank you very much if you type
4.5
that in.
approximately. That's 4.5.
All right, we're off to find something new. Oh yeah, the big fan.
That big one out there.
Pilots,
at
we're looking to get
noon.
that move back underway, not the
That's right.
whoopsie
Calling us.
We've had this target.
That is
Co-pilot's
huge.
ready.
Very candelabra shaped.
I'll put in another 20-meter move
That
then.
is a big, big
Copy.
gorg.
Bridge, this is
Are you
RB nav.
thinking from this
We have
distance?
a
I'm going
move
to
if you're
guess
It'll be the same as the
coral,
last.
just
20
based on
meters, bearing 270
the rings,
degrees,
the-
speed 0.2 knots,
the
please.
base.
To see some.
That's at least a meter.
Good copy. Thank you very much, Bridge. Pilots, that move is underway.
Copy nav.
Nice.
All right. Get a look at these.
A rhabdoplectella.
Oh.
Hey, sorry I've been on a call, but I saw this, and this is really exciting. So This is a bamboo coral. This is, we'll have to see it from another angle, but I think this is the candelabrum that Les Watling and I are working on describing right
Oh,
now.
nice.
Well, that's great.
Yeah, so this would be a new genus. If it's what I think it is, it's what we call clade I4.
Okay.
You're being very careful to make sure you don't ruin the name by using it.
Yeah, I don't want to the name. Yeah, no nominutums here. So what we'll be looking for is to see where it all comes together. It should be like one stalk that comes off the ground, and then you get a point where the branches sort of spring out. And is it a trident in the center there with branches extending from either side of the trident?
Okay, so you want a good look at the main stalk.
Yeah, the main stalk where the branches start.
Okay.
Tilt down when
Great
you're ready.
view. We're going to be able to look at this also to learn something about the sclerites, which are these small calcium carbonate microscopic bits that are embedded in the soft tissue that help create support and possibly protection against some predators, although there are plenty of predators of octocorals that seem to chew right through them. But those are very widely used in the taxonomy of octocorals, both for naming a species and for identifying them later. So this exquisite video that we get from the Okeanos Explorer helps us with that to some degree. Ultimately, you need to have it under a microscope.
Yep.
Very interesting, if people realized what is used to identify these things, and if you were looking at, like a field guide to the corals, most of it would be filled
say
with
I'm
scanning
definitely
electron
moving now.
microscope images of microscopic bits and not the level we're looking at here,
So it's really
which is
good
why it's so hard for us to
follow up
make
in
an
the center
identification when we see it on video. We're still working at combining those two elements together.
Right.
Same with the sponges. I mean, they look.
Oh,
Some of
yeah,
them look
sure.
exactly the same, and they're in totally different orders, and it's crazy.
So one of the things you see as we're panning down here is that these upright vertical branches don't have sub-branches. So that's one of the clues. Instead, it'll be like a candelabrum. They're all coming off of kind of like a base of a laterally extending branch, so to speak. And the older they get, the more of those vertical branches they add on to the sides.
Nice.
Yeah. And you can very clearly see the white calcareous internodes and then those black proteinaceous nodes through the tissue.
That's indicative of bamboo coral.
Yeah, so these ones, I do not see any of the needle-like sclerites poking between the tentacles, what we call intertentacular sclerites. They're probably there, they're just not so prominent as to be poking up between them. All right, so there's the middle point. That looks like a triple junction, doesn't it?
Yes, it does.
That's what I'm looking for. Beautiful, beautiful. This is it. It's a great find.
Nice.
Go ahead, Bridge.
Thank you.
Yep.
Good copy. Thank you very much.
I was wondering if we need a sample since it's
Pilot
so
side move is complete.
relatively new.
Thank you, Nav.
Yep.
Really, really nice. And did we get the lasers on that? I think they were, yeah.
Yeah.
Oh, that shot. That's going to be a nice
It almost looks like a bird leg with a stocking on it.
Yeah, it looks a little wrinkled.
right down.
Yeah, when you see them with these sort of thickened and swollen nodes, you know they're really quite old. And what's happening is the deposition of the calcium by the soft tissue is starting to grow over that proteinaceous node. So the farther down we get, the more it looks like a ring that's curling over. It's almost like, you know, when you see trees that grow into like a barbed wire fence that's next to it, it kind of girdles and grows around it eventually. So eventually, if we came back in another 50 years, we might not see a node there at all, but we'd still see that knobby knee swelling.
Okay.
We're close up on that stock. We also saw ubiquitous hydroids, which are coating the surfaces of everything, looking for small prey.
And can you paint it with lasers when you get a chance, just so we can get a good size?
Tilt up.
Yeah, what a great view. It almost looks like it's one half of the candelabrum is growing really well, but the part closer to us is not. That part has been broken off, and I'd say that this colony has been stable on that rock for a very long period of time.
Yeah, for sure.
Yeah, you can see the blue sponges growing on the rocks there behind as well.
All right, come lasers. Yeah, so lasers are 10 centimeters.
And also try to run it across those.
So the candelabra part of the base is at least a meter. So that would
between the
put
node
that
and the main
stalk at
stalk.
another meter.
2 meters tall at least.
Yeah.
Yeah, if you can, it's not terribly critical,
Yeah,
but if you
rotating
can,
down,
when you're
keep your
moving, if you
view,
could get a
have
face-on
plenty of
view of that
time.
candelabrum, that would be so awesome. But
So you want us to swing? You want us to swing left, so we can get a view
Yeah,
from
that'd
the, like a more of a front facial. Oh my God, I can't. I cannot talk today. Yeah,
A frontal view.
a frontal view. Left our side, 9 o'clock. Yeah, it's like swing
Now,
9.
did you copy that? Pilot asked how much
So
is
we can
left
see
of
all
the
the
node.
individual branches.
The ships have been stationary for about 3 minutes now.
I think here, if we get that, you'll see that it's going to be lopsided. So it's growing off to the left, but not to the right. It should be adding those vertical branches symmetrically around the trident. The center part where the branches appear to begin.
Awesome.
Very nice.
In the middle upper.
So we're still below 1,400 meters.
Yeah, we haven't moved very much at all. We got somebody out there.
And you know, what we're seeing is really characteristic of 1,400 meters. So this is looking a lot more like, you know, offshore seamount at this depth than it is when we're inshore on Lake Plateau and seeing the Lophelia mounds. We're getting a different community, a different representative group of corals.
Absolutely.
Really good to see. Thanks so much for that imagery. That was exquisite.
Yeah. We're good whenever you guys
On
are.
the other side.
It's just so much to look at. Not sure what's going on.
One more pirouette. We're going to go around the other side.
So to the left there might be another bunch of those hermit crabs again on a dead skeleton. Isn't that interesting?
Yeah. Similar to the one before.
Yeah, the ship's been stationary for about 5 minutes now. Should be good.
So I'm curious if this mound is not biogenic, which it does not appear to be, although we still got a ways to go to get to the top.
Mm-hmm.
But then how it might have formed.
I have no idea.
Yeah. Maybe there'll be some geologists in the chat
You can chime in and let us know.
could either call in or type in.
Oh, neat. Here's the backside of it.
Mm-hmm.
Kind of a cool view. You can
Yeah,
see that all the
looks
polyps
clear
are
to the tether.
concentrated on one face.
Mm-hmm. I would assume that face is
Question is
facing the current.
Well, I was just going to say, Stephanie, is that the downstream or the upstream
Yeah.
face? So, and some of them, it's downstream because the water
Maybe
flows
the next
through.
one turn
The
into going
baffling
to
of the
go,
branches
but then
causes
snap
there
at
to
the
be
back.
these little ripples behind it.
Okay.
And so maybe it's
Rotate
better to be on the
down.
downstream side
Right.
than the upstream side. I'm not actually sure what it is for this
That's
one.
71 now.
Great view here, though. As we are zooming in, I can really nicely see the original trident point. And here you can see what I mean about each subsequent vertical branch starts as, this curvature from the existing one at the next node. And normally that would be symmetrical around the original trident starting point. And the hypothesis
Go
here,
down
we haven't
from
been
the
able to
side.
prove it yet because we haven't had the appropriate samples, but the hypothesis is that when it's really young, it's growing as a whip. You see that it has to get to a certain size before it starts putting out these side branches.
Right.
I've never seen one that's really short to the ground and grows this way. So they all, have that center stalk grow for, let's say, 50 centimeters to a meter before they start branching. Just incredible.
Awesome.
Thanks
Good
for
stuff.
flying.
Yeah. Really good.
Video's clear.
There's some more Sticopathes at 9 and at noon. Looks like we have some undercut ledges a little bit.
Another Metallogorgia. A Metallogorgia.
Metallogorgia?
Yes.
So the next thing we have to train you for is finding what we call the teenage Metallogorgia, because they only have that sort of terminal parasol as their final growth stage. As they're growing, they have branches come out along the stalk, and they don't have anything at the top. It looks like just a little antenna. But then what happens, we don't know why, but when they get to this phase, they drop all the branches along the stalk.
Interesting. Looks like there might be something dead here at 3.
We could probably put the move in now.
And then in the center
By the
we've
time
got
that starts swinging, you'll
Is that a
be
dead whip?
out of the
ophiuroids all
out of
over
the-
Lots of
something.
ophiuroids?
All over a whip or...
I'll bet it's a dead Metallogorgia.
Oh, dead Metallogorgia. Yeah, you can see the branching
Yeah.
at the top.
Right, and so this is instructive also because you see it doesn't have the Ophiuchreus, which is the brittle star that lives associated with it. And on the live ones that did have the Ophiuchreus and the purple tissue on it, you didn't see anything on the stalk. And so they tend to keep associates off of it. And I don't know if that's just the Ophiuchreus ophiuroid that's living there to sort of swats things off from settling on it, or if it's some secondary chemical that the coral is producing that is noxious to anything settling on it. But you only ever see stuff on the dead
Right,
skeleton.
pellets. Direction to
So,
waypoint
very
2 is a
instructive
242 now. Keep that in mind as you stretch out, but we can
Okay,
follow
so
the
it looks
local
like
high
these
if you
rocks are
see it
starting
at
to
a different
increase in more of like a
heading than
piling of boulders.
242.
They're about 1 to maybe 2 meters wide, maybe half a meter tall, with sand in between.
Rotating up.
The slight rise here, I would a very slow slope coming up,
Copy.
maybe
coming
10
about.
degrees looking ahead.
Yeah, we can call it 240 for now.
Auto heading set for 242. All right, Arrow.
Another bamboo ahead, it
Gotta
looks
slide
like.
up
Different
a little better.
species here.
And rotating up.
Oop, there's something in the water. Quick,
Yeah.
idea, quick.
I'm at 4.5.
It had tentacles. Yeah, you can really see the current moving across.
Copy that. We'll do 20 meters at 240.
There's another bamboo,
Copy.
I think. Maybe not.
Bridge, this is RV Nav.
Might be something else.
We have a move when you're ready. It'll be a 20 meter, 20 meter move at a 240 degree bearing.
This is
Speed
the
decimal 2
Primnoidae
knots, please.
No,
Mhm.
okay.
This one may be different from the one we looked at earlier, and I'm basing that on the orientation of the polyps pointing downwards.
Good copy,
where I
Bridge.
believe
Thank you
last
much.
time they were pointing upwards.
Pilot
This
set move is
could
underway.
potentially be the genus
Copy that,
Narella,
thank
but
you.
I'm going out on a limb there. Let Alan talk about the hydroids there. Alan, is there any hope of identifying hydroids in situ, or is it just too difficult?
Yeah, probably too difficult, other than, maybe getting to a family or something like that.
Yeah, there's something yellow here at 10-ish. And this looks like a bamboo coral at 2-ish, the tall one. Yeah, that dead center pink thing, that'd be cool.
Suggested flying by.
Maybe it's his Bathypathes, maybe? Yeah. So it's maybe Bathypathes bipennata? Type of Antipatharian. That's my best guess on that guy.
Yeah, this looks like Bathypathes, a different kind of black coral, a different genus now.
I got it
All
right.
crowded in together.
Yep. I like how all the black coral generic names seem to...
Pathes.
Pathes,
Yeah.
yes.
Makes the life a little
Tilt
easier
down to get a
when you're
full
learning
screen.
Latin.
That'll also get the other two corals in the shot.
So they thought they...
Yeah, and I guess the factoid, we have to give this factoid out every expedition, but they all belong to the Antipatharia, and Antipatharia translates as against disease, and it's based on this old Greek idea, 2,000 years ago, that crushing up the skeleton would have medicinal properties, which it does not. That's where the name comes from.
There is a really cool spiky ball right off screen when you guys are done looking at the Bathypathes. It's
Top
right
of the
above, like at 1 o'clock, right off screen. There's this really weird spiky ball. See, it's at noon.
Right
Just
at the
popped
top.
out.
And I think the white thing there next to the black corals, that's a corallium that's standing upright. We looked at one that was knocked over.
Just tilt down to get the coral.
Or hemicorallium, corallium.
There's also a single-stalked Antipatharian
coral.
here.
He just described it.
Hermit crab climbing
Yep,
up on
this-
top of that.
Same pink hermit crab.
Corallid.
And there's also some kind of legged thing, pink, maybe a sea spider, pink sea spider in there.
That's max zoom.
I think that's what it is. I see his little legs.
Look like he's got.
I
his
wonder if it's
proboscis
feeding
in.
on that corallid. Be nice to see it
It munching.
munching, exactly.
Munch.
No offense to the corallid.
Oh yeah, it's not personal. And maybe a Chrysogorgia too at 3.
Great. Wow. Huge rocks.
Yeah, this is a big boulder. Maybe 5 meters wide.
Sight of that spiky thing. Oh, I see it.
Go ahead, Bridge. Good copy. Thank you. Pilot's
It's
move
so
is
interesting
complete.
to see this community because if I didn't know better, I would think I was on an offshore seamount and not on a sedimented continental slope.
Mm-hmm. There we go.
Oh, yes.
Too cool.
You can see it. It's got its
What, proboscis?
proboscis there, and it sucks the juices out.
So there's pink on this coral. Is that coming from it being traumatized?
I don't
From
know.
being
That's
eaten?
an anemone.
It's
an anemone.
oh,
growing on
anemone.
the coral.
Okay.
And it's unfortunately been attracted, or it has attracted this sea spider to suck its juices out.
That is such a cool sea spider. He's bright pink.
Yep, filling up its branched stomach, which descends down into its legs.
Yeah, very cool. I don't know how many people realize, Alan, you described that well, the sea spider and that long proboscis and the
Full
suction,
screen of the
but
coral.
most spiders suck their food. They liquefy their food before they ingest it. And the sea spider isn't that closely related to terrestrial spiders, but terrestrial
Slug
spiders do
tilt
something
down.
similar. They basically pre-digest outside the body and then suck it up.
That's really cool. Predation caught. Now I wish we knew more about that anemone and what it actually looked like when it's out and happy, so we have a more specific. And all these hermit crabs are climbing up on things.
You got something neat in that hole up there.
Between those two stalks.
Yeah.
Might be a crab or something.
Also something
And we have
interesting,
that spiky
possible
ball
spot
too up here
lobster or
at 1.
Something inside
But
the
it's tiny, but. Oh, that looks like a coral or something? Maybe
Oh.
a solitary coral?
No, it's an
Or 2.
octocoral.
Oh, yeah, it has 2 polyps. It may be a pink Mesodacea or sponge above it.
I'm sorry.
That's probably Anthomastus or
Anthomastus.
related to Anthomastus, pseudanthomastus, something like that, which is an octocoral which does have these relatively large polyps. They kind of grow a little bit like a toadstool,
Rotating
and
down a
the
little
polyps
bit.
would normally be coming out of that, so this would be an early stage. If
Ship
that's
has
what
been
it is.
stationary for about 4 minutes
Very
now.
unusual.
You should get
Actually, yeah, they're
be
two
closely
polyps
under
there,
your
aren't they?
spine.
So that's
Go
almost
pilot.
really what it is,
Rotate
just
right
a
for
relatively
that
decent recruit.
And these yellow hexactinellid maybe?
No,
No,
definitely
yellow?
lemon sponge. Yeah.
I think these ones
Spiky
Yeah.
right edge.
The orange demo sponge, and then tan right to the right of that, and then on your way to your spiky ball. Which,
I don't have a clue what that spiky ball is. Sponge.
yeah, I'm thinking that's a sponge.
Baby sponge.
Maxim.
Maybe a hexactinellid in front of it.
Possibly a small. Yeah.
That's neat. Little starburst sponge.
Really
That's
great.
pretty wild.
Definitely very unusual.
Okay, we're good whenever you guys are. You can see the dead stalks here. Ooh, we got
Yeah, it's very surprising.
a squat lobster at noon. And then we had a yellow sponge to the... Okay, pilot change, no problem. Yeah, there's a lot of stuff down here.
Yeah, we're still at 1,400 meters.
We haven't moved at all. How much longer do we have in this dive? Let's see, 3:45? 2 more hours. So the goal is to get to the top in the next 2 hours.
Yeah.
We got plenty of time.
An hour and a half to get to the top.
What, John?
I think you have an hour and a half to get to the top.
Hour and a half, yeah.
You got to go.
You got to be off bottom at
We got to get up there.
5:30 or 5:45?
I think it's
5:30? 5:45 we're coming up?
5:45.
Yeah, this yellow thing out. Yeah, there's a white thing in the background at 9 I'd like to take a peek at. We're just not going to go. Just sit down. We're just going to stay here. Just look at all this stuff on this one rock.
It's just so rich. You know, it's not really dense, you know, like you see with the coral mounds, but there's so many different things, and there's at a lot of different scales. It's some.
I noted before, since we're sitting here, that bamboo coral to the right is different from that candelabrum that we were looking at.
Oh, yeah,
earlier.
definitely. It's totally branched different. It's internodal branching, too, whereas the other one was nodal, right?
Hi, yeah, excellent. So it's probably the genus Keratoisis in the genetic clade D. Had a tough life. It's missing all the tissue from the lower part of the colony. So
Yeah, right.
may have had
Looks pretty beat up.
encounter with a sponge at some point. Excuse me, with a sea star. Star at some point.
But live to tell the tale. Via.
Yeah. We actually have our eyes on something at 9 o'clock when you guys are done, so don't drift too far away, please. Hey, Scott, can you type Keratoisis into the chat for me?
Nav, Chris will be right in for you.
It's
Copy that.
Worms hates me.
Hey, Pat, so I'm looking down at 72 degrees.
Yeah, this yellow
Cam.
sponge we are after right here
I
at,
just thought I might have a
It's
little
at
bit
6
of
o'clock.
Keera.
Thank you.
But don't see any concern up ahead.
Oh, Keera.
Yeah.
And I see a sea star, I think, right below it. Maybe a goniaster. So this yellow sponge, it's got lots of film on it.
That's
Like a bacterial mat on top. Kind of sickly. It's pretty neat, though.
yeah. It may like it like that.
Yeah.
You know, we often see what we refer to as these deep-sea spider webs. We find them on overhangs and so on. And just recently I saw a paper that goes, oh, maybe this is what I'm seeing. And that is the sperm release from some of these sponges comes out with all this mucus. I'll try to find a link and send it to you, but Alan, you may be familiar with it. And I'm wondering if that's what a lot of this sort of webby stuff is. Evidence that there's been this spawning from these sponges.
Possible. This almost looks like it has a fistule on top here. This, like, sharp. I think that's part of the sponge.
I would guess not, but I really don't know.
Yeah, that's neat.
What is the structure that the sponge is growing
I think
on?
it's a dead coral.
Oh, with. Okay.
That's a guess. Old dead. It's very old. You think? Look, it might be a dead old sponge. Yeah, and then we've got this little star here. It's also a single-stalked gorgonian of some type.
Extra lights coming on. 3, 2, 1.
That's much better.
Good eyes and
So
another
that light's a
small
little
white
better, yeah.
sponge on the left-hand side there.
Let's look at those. That lower right.
Are we putting this in the demo or the hex?
That's definitely a demo sponge.
Okay. Maybe a worm tube? Yeah, that looks like a dead coral.
I'm saying a sponge stock.
Sponge stock. Is that another hermit out there?
Yeah,
Or
another pink
all
hermie.
these Lots of hydroids colonized on this dead coral.
For the base, not that.
No, I thought there was something in the distance.
Like tree branches.
Yeah, it's
I mean-
crazy. Yeah, we can move on, I think. We got a good look at most of this stuff.
Yeah, we got to keep moving.
Mm-hmm. Not too fast, though. We got 2 hours to kill.
That's true.
And like, I don't know, 100 feet to go. How far is this?
We can still rise another 70 meters or so
Okay.
to get to the top of this. Do you think this is
white
a
ball's
convenient time? Yes, this white ball is great and would be great to get a zoom on it. That we should get our middle of our transect eDNA sometime soon when we.
What was the depth? Why don't we wait till 50 meters up?
Okay.
Because it's a 100-meter difference, right? So if we came up 50 meters and then did it at the top, 100 meters.
Right. So we'll want to do it at about 1380.
Yeah.
What do we got here?
Ooh, Oceanapia, maybe? Oh, yeah.
Yeah.
I guess is Oceanapia on that guy.
Right
but-
next to a Pharea, a glass sponge in the background, but this is, wow, just a
That's
beautiful
super neat.
demo sponge. A morphology you don't see often.
So I didn't catch the name. This is the thing I always call polymastia.
Oh, polymastia. You're right.
That spiky one we saw earlier was a small version of it. So what was the name?
Polymastia. You're correct. That's right. I was thinking Oceanapia, but you're right. It's polymastia.
I'm going to have to trust you guys on that one.
We don't usually come up with the correct sponge name. So that's
Oh, yeah, no.
Thank you.
You're right. We got some worms, too. Some feather dusters here at
Oh,
here,
yeah.
right off screen, right at the edge of the screen.
And a nice blue sponge tucked underneath that guy.
Mm-hmm.
Video's clear.
Oh, there's some Sargassum detritus. So that's just drifted down from the surface. And then maybe an anemone. Yeah. Again, it's like this pile of boulders with sand in between. The boulders are 2-3 meters tall, maybe.
Gonna
I mean,
volley
wide,
up and
and
see
maybe a
push
meter
out.
tall in
I
parts.
think I'm all done moving.
They're kind
I
of
don't think
jumbled, like a pile of
settled out.
rocks.
Cam set at 5-0, please.
So I just heard in the chat that Bruce Mundy ID'd one of the fishes that we observed yesterday that we
Okay.
were unable to identify as a juvenile lancet fish.
Oh,
And
cool.
that was a very unusual observation.
Video
So
ready for fog
Oh,
lights.
nice.
that was
Turn
very
off.
nice that we were able to capture that. It went out, and people were able to
Looks like you're cracked off
use
little
use that
rotated.
high definition video and to create more knowledge.
Yep. Give us some feedback.
It's perfect example of this telepresence and how well it works in these NOAA
Community.
ocean exploration expeditions.
Yep, community engagement. There's There's a little white stalk here at the center. It's probably
I'm looking at you
something
45.
has
Can I
passed.
get another
Dead.
20 meters in? Just to get some moves in, or do you want to wait? Yep.
Another stick of pathies, some dead stalks.
Copy that. I'm looking 250.
See these big piles
20
of boulders.
meters,
I
250.
just want to know where they came from.
I know.
So we're keep that.
Where did you come from?
Okay.
It looks like, so when we're on the,
On
in
the
the
way, point
MPA
two.
with the same kind of similar, like, squarish boulders, and they look like, almost like they were formed from the
Canvas at the
pavement,
3-5
and then
degrees.
underneath collapsed.
That's nice. Get a
Hey, a
little,
little rat tail.
little.
What, Nizumia, maybe?
Really nice. I think there's a. Is that a gastropod on that? On that dead whip? Oh no, that's another.
A hermit.
Another hermit that's climbed up, and another. But there's a fish to be seen.
Mm-hmm.
Oh, so cool. More Metallogorgia.
Violet, can I look around with your blue view for a second?
Yeah, it looks like that bamboo coral's dead. There's just the stalk left.
Yeah, and I think that's a candelabrum one as well. You can see
Mm-hmm.
the trident in the center again, so it's informative.
Mm-hmm.
When I say informative, what I mean is we're still learning about the distribution. We want to know, where it's found. We've found colonies in the Pacific and the
Sure.
Atlantic, and they seem to be within a certain latitudinal zone, so tropical to temperate, but not further north.
At 3 o'clock, we have a fish in the sand. And then above that, we're going to have something yellow on the rocks.
Are the lasers still? There they are.
Who's joined us on the line?
What this guy is.
No problem.
Oh, he's neat.
He is. Very beefy eel.
He looks kind of beat up. Like his skin's sloughed, right?
Mm-hmm.
I don't know if that's normal or not.
This will be someone's.
Cuskiel, maybe? Oh, he's got little feelers.
Yeah, he does.
He probably doesn't see very well. It's pretty deep.
Going to the eyes. Max zoom.
Is this a conger eel or a cusk eel? I don't know.
It's a fish. That's the best I can do. It's a ray fin fish. That's where I tap out.
Whip right
That's all
ahead.
right. Great footage of it. Should be very easy to identify for someone who knows what they're doing.
Look at that beautiful sponge at noon.
Oh yeah, let's check that guy out. Cusk eel is what.
Cuskiel? Okay.
Well, that's what Scott wagers.
Oh, here we go. Kevin said it has feelers and it's pelvic rays. That's neat.
Chidiformes.
Ophidoformes.
Ohidoformes.
Ohidoformes.
There we are. Ohidiformes.
And if we could glance to the left, Steve says there's a lyrate colony we haven't seen before.
I'm not sure. What kind of colony?
Go ahead, Bridge. Roger that, thanks.
Black coral colony? It might be what they mentioned.
Move is complete, pilots.
Copy that.
So yeah, behind the Sticopathes is this orange whip in the front, and then there's another black heart right behind it. It has gray polyps. Really loose branches. I think that's okay. It's that orange sponge I'm interested in.
Yeah, the black coral, I'm not sure if that's a young Leopathes.
Leo, okay.
Or if that's something else. It's definitely new for the day, though.
Nice.
Now we're going to come get a look at this sponge.
Oh, neat. It's like a skirt, like a fan skirt.
Oh, this could be the glass sponge Hertwiggia.
Looks like on top of that rock we got another flopped over coral too. There we go. This is bright yellow. And you see the veining in it?
Yeah. Yeah, I don't think that's Hertwiggia. I think that's a demo sponge.
Always more zoom.
Nice.
Still I see the
Very
atom.
nice footage.
So this is a yellow, I'm just going to call it a yellow skirt sponge. Or amphitheater. It has veins. All right, sounds good to me.
Thanks for getting that. Nice view of that yellow sponge.
So again, we're looking up slope here.
Okay.
Our midway point is going to be about, what, 1380?
Yeah, about 1380, in which
Okay.
case we'll take another water sample.
I got something here as a fish, maybe? Another rat tail at 6 o'clock. Oh, he's in the breeze. Oh, Lemonema. A coddling of some kind. Look, there's Feria in there too. Yeah, we're good.
Hey, Chris, looking, it does look like Niskin 2 is shut. If 5's okay. Could we do a quick zoom? You just stay right there.
Looks like a funky
That's
sponge
really good.
here at 6. Possibly dead. Can we look at that coral at 6 o'clock? I
something
think
in it?
the's something
Yeah.
in it.
Is that trash?
So the second one did
I hope
fire.
not.
Me too, but it would be good
so
to take
2
a
and
look.
3, I think, we
Yeah.
did simultaneously.
It's going off
Oh.
screen. There's a coral going right off the corner
that's good
screen.
video.
Yep,
Thank
on the
you.
right.
Zoom out.
And it looks like maybe black trash inside that coral head. I just wanted to get a better peek. I also think I see a giant star in the background. It looks like trash to me.
Where's
Ugh.
that?
That looks like plastic.
Very sad to see.
Ooh, there's a cool, right underneath it, there's a giant crinoid. And then off in the distance at like 3, there's a different starfish or sea star. There we go. Now this one looks similar to the other one we saw, but this one doesn't have those long pinnules at the tips. I think the other one was Analceta metra. This one looks like a different species. I think there's 10 arms. Hi, who chimed in?
Just me again.
Hi, John.
Looking pretty settled out, pilot.
This is a type of cumatulid.
Camera's at 45 degrees.
Sea lily is the common name, right?
Sea lily, yeah.
And if Chuck Messing was on the phone, he'd be able to tell us from the footage what it is, but...
It's a beautiful crinoid is what it is.
Yeah, it's pretty cool. So they reach their arms out and they catch food in their pinnules, and then they pass it from one pinnule to the next all the way down into their mouth. And their mouth is right there in the center of that arm, those arms, just like most echinoderms, right, relative to the sea stars.
And what is a pinnule?
I was actually watching something, again, that PBS special, right? I was watching that PBS special where they were talking about how bilateral symmetry is actually what gave rise to all the ability for, the evolution to take place, right? So lots of variety in that bilateral symmetry. And the original, the oldest echinoderms that we have fossils for were actually bilateral. And they formed radial symmetry over time.
Yeah.
So that was pretty neat.
They are.
What is a pinnule?
Yeah. As someone who
What's
studies...
the pinnules? The pinnules are the little arms off the arm. It's like the subarm, right? So, sorry, go ahead. I didn't mean to cut you
I was
off.
just going to make a comment about how there's a lot of diversity in the non-bilaterian animals as well, but I get the point.
Yeah, because like the first animal fossil we have, the animal actually looks like a plant. Like it's a long stem, and then it's almost fractal.
Yeah.
Right? Where the tissue fractals into these. Yeah, you really have to just watch it.
The Ediacaran fauna. Yeah.
I got a sea star coming up. My guess is some type of goniasterid. And a little blue sponge at 3. And again, another Anthomastus past that at 3 o'clock. Yeah, let's get a good look at this sea star. Yeah. Some type of goniasterid.
I'm falling down and I can't get up.
I know, right? It's probably, yeah. Looks like it had lost an arm maybe, regrowing an arm at the top. It's a little shorter than everybody else.
Looks like it's eating.
Could be eating. Maybe eating that primnoid it's on. It looks like the tips of his arms have like almost like a forked hook.
Yeah.
That's cool. Goniasterity. We're good whenever you guys are.
You ready for another 20 meters, pilot?
Keep your eyes open for a dead Lophelia. John Reed saw one.
Oh, where?
Yeah, it was several minutes ago.
Okay.
So we're a little
About maybe
past.
Looking like 245.
No more than 10, 15
245.
centimeters.
Oh, little guy.
It looked like the
20
typical
meters.
old dead black
good to me.
Ophelia.
Okay. Was it standing?
Branching, I could see polyps, but we were beyond it before I had my phone died on me, so I wasn't able to call in right. So anyway, it appears to be around here.
Okay, I'll keep an eye out. Looks like we have a couple fan sponges and some maybe white corals, some gorgonians. Now that looks like Ficalia to me. See the veining?
You'll want to look around to the right of it.
Yeah, on
So
the
veining,
top one.
Yeah, the veining on this might be on the other side. If you face looking at its coming in from the right, there might be some fanning. Oh, it looks like a sea pen on a rock in the background too.
Good eye, Stephanie.
On the right-hand side.
Now you were saying these are important. You want to collect that?
So, I mean, that would be so fantastic. We should get a good image of it at least. What I can tell you is that there's only a single, maybe there's two now, because maybe Okeanos collected one 2 years ago, but as far as I remember, there's only a single documented collected rock pen in the Atlantic. All the others are in the Pacific. And that one that was collected in the Atlantic was collected in the Bahamas on an expedition we did back in 2009 and is a species known only from the Atlantic, and it's the one that's in a different genus from all the others. And when I was discussing yesterday why that collection was so important was to ascertain, you could see this, doesn't this look a lot like the second sequence we collected yesterday?
Yeah.
Mm-hmm.
Sediment? Yes, that's what we were saying, that overall colony morphology looks similar, but here you can see that it's kind of like a bathroom plunger or suction cup, the way it's held onto the rock. The peduncle has been modified to cling onto it in that way. So that's what's different about the rock pen.
So this we would call this a collectible.
Do we do easy stop? If we're gonna collect?
I would.
Yeah. Can we collect this? Can you guys get in there deep enough to collect that?
So if you're going to consider that, let me say that it can be grasped in, the Tygon tubing part of the claw, and then just kind of wiggled, and it'll pop free of the sediment.
Okay.
Pop free of the rock.
Sure thing.
And it'll come off pretty easily. It doesn't need to be cut.
Sure, we're fine. But
Okay.
That peduncle is critical. We really like to see that.
Yeah. Did you guys hear what Scott said, or should I repeat for you? Okay. So the rubber tubes inside your claw.
Okay.
If you can just gently, without actually cutting it, just gently squeeze them with the rubber part, and then give it a little wiggle. He might just pop off that rock, because the bottom part, the peduncle part, is actually the part that's really important to maintain the structure.
do you want to put your mini Zeus up at off or position?
I can keep an eye on Sirius.
Again,
if you like.
you see this inside that
Sure.
fan at 9. You're getting that veining.
Mm-hmm.
So that's probably Ficalia. This is going to be sample number 5. If you can get it. You don't think you can get in there?
I mean, could you suction it up?
Yeah, that's kind of what I was thinking.
It might be hard for the jaws to get that far.
Maybe a
to the rubber.
Chrysogorgid there at 9 o'clock too. That
Just pick the whole
furry.
rock up.
Yeah.
Yeah.
Yeah, if you guys can't get it, it's fine.
Collectible, it's okay. He's really happy with the footage.
Can we get the lights on?
Forward lights coming on.
Up until yesterday, I did not know of the existence of rock
Right?
pens.
Yeah, because they're always
sea
in the
pens
sediment.
Yes.
So this is the one weirdo from the
Look at the base.
Yeah,
family. He's the black sheep.
Max zoom.
Mm-hmm. You can see the inside.
Mm-hmm.
It's like tissue. Almost muscular, like that,
Oh,
like fibrous.
yeah. It looks like the part that's under the sand, usually, only.
Yeah, you don't usually get to see it because it's underneath that,
And then
that
it's
sediment.
modified to have this
polyps in the way.
adhesive foot. It's pretty neat
Okay, let's go full screen.
adaptation.
Alan, you said
know
adhesive. I mean, I don't know that it's
what area
not
you're going for?
adhesive, but the descriptions based on the collected samples in the Pacific suggest that
Okay.
in the center there, you can see at the bottom of that rod, they use musculature
Starboard
to pull up
outer.
the
Starboard.
middle center
Starboard
of
outer's kinda
the
tricky.
Suction.
to create that suction.
Hey, Scott, do they move around?
Gravity, though. Right.
What's
That's
that?
great.
Not
Gravity.
to my knowledge.
Usually once they're down,
You know, I've
they're...
also
point.
tried them-
Port outer's
you know those
taken,
clowns
correct.
that you can sort of box with that sort of fall over and then bounce back
Okay,
up?
yeah, with the
I've described
step.
them that way. So we have seen them become dislodged and they drift for a while, and I suppose they can
Yeah, it's up to you.
hopefully upright themselves.
reattach.
But, I mean, they're not mobile.
It's not like an anemone that'll kind of like
I guess
crawl
I'm not
around
really
the
sure how
ground,
big
you
it
know?
is.
Not to my knowledge, but
Okay.
you know, we don't know all that much about them. There aren't all that many observations. I will say that we found an observation in the Pacific, not too far from
Yeah.
Hawaii, at about 350 meters,
Copy.
where we saw these huge fields of
You try to
literally
scrape this out of the
thousands
box,
them.
too,
And
after you
that
grab the
was
rock.
just
I
an
don't know.
incredible sight.
Nice.
It's up to you.
But otherwise, yeah, we normally
I
just see
support
them
you.
in ones, twos, and so on. I say normally, like we've seen a
Want
lot.
me to center sample
We
up a
have
little?
not seen that many.
Well, good eye spotting
Hold
that guy.
there.
Yeah, he's pretty small. Move the rock first? Yeah. See if that's loose.
Can you get the whole rock?
That's what I was thinking. Just grab the whole rock. Stick it in the rock box. I'm not sure, John. It looks like that rock's pretty solidified in there.
Yeah, it looked like that rock's a boulder.
Yeah, the boulder.
Do we have a taxonomic name for this collection?
Besides
Besides-
Peninsulacia?
Yeah. Thinking that Upsana says in the chat maybe Anthoptilum or
Yeah.
Calibolumnon.
I mean, you could
Yeah, Anthoptilum or Calibolemnon.
try for
so
that
the one in the
inch or
Atlantic
so before the
is
blades,
currently
but
Calibolemnon,
Okay.
And then
and the ones in the Pacific
Oh, yeah, you
are
could do that.
make sure
Anthoptilum.
the cutter's still
your
above
choice.
it. And
Great.
then try to pluck it.
We're going to go with the Atlantic
You're
name,
only getting,
right?
Yeah,
part of it.
that's what I put in.
But it still might be tough to get all the way in there.
Thanks for that,
Jaws are pretty long.
Scott and Upsana.
Go.
vehicles look good.
Calibolumnon.
Looks
I
like you're all
it.
steady, Sean.
Copy that.
Looks like a little wishing bone behind there. Little rock, little branch thing. See it? Little deep sea wishing bone.
Yeah.
Max zoom on.
Oh, it is small.
Yeah, it's pretty small. It's like 10 centimeters long, maybe. Yeah, just gentle, yeah. Whoop, popped right off. There you go. You got it.
Nice.
There you go. Peduncle's intact.
Oh, look at the
Do you know where you're going with this?
Look at the little ringlet left. It had to have been there for a while, huh?
Yeah.
That's pretty neat. Good close-up shot of the peduncle. Beautiful. See, you can see it like turns into a suction cup, right? It
Yeah.
sticks to that rock.
That's a really cool way.
Like a like a bathroom toy. A bathtub toy.
That is such a good view. That is not a view that we get in situ ever. Fantastic. Nice work, pilots.
All right. Ready to open drawer. Extended drawer.
Now we get
Full
to watch
extension.
it go into its container.
Coming down a hair on
Port.
noses. Stopped.
Port inner.
That one is going to be number 5, yeah.
Great. 5B.
Sinking. Retracting.
Nicely
Nice
done. It's in the box.
Nice sword.
So I was just realizing
nice
the
grab.
increased value, particularly of this specimen, relative to what we did yesterday, because these are two locations that are quite close to one another. So this may be the first instance where we have, within 30, 40 miles of each other,
coming in.
two colonies
Two
that
rock
look the
prints?
same, one in sediment and one in the rock.
Oh, yeah, cool.
So that's going to be really interesting to compare.
Awesome. Thank you.
Oh, and I just see in the chat the Calibelemnon species in the Atlantic is named after you, Scott. Is it Calibelemnon
That is correct,
franci? That's quite an honor.
Yeah, that was a real surprise. Gary Williams, Kyle Academy.
That's great.
I'm looking 50 degrees.
Nice collection.
That's like a white gorg noon or 1. And a little yellow one next to it. There might be another Bathopathes in the background. Yeah, some kind of Gorgonian. Seems pretty disorganized.
Mm-hmm.
Planar.
So that.
Yeah, I'm going to go again with Coralliidae for this one.
Coralliidae.
Hemicorallium or corallium. I'm not sure if Steve Oskevich is still on. He might have something to say, but that kind of looks like what it is. I'm seeing lots of these smaller bumps behind the polyps. Those might be, what's the word? Siphonozoids, a secondary polyp that is non-feeding, really small for funneling water.
Mm.
It's either that or it's an Anthothelidae, but I'm pretty sure it's Coralliidae, in which case it would have a very hard, brittle skeleton.
Mm-hmm.
Go full specimen.
Man, I'm learning so much on this.
Yes, please.
Lights coming on.
Let's get another.
Yeah, see these white, this like clear sponge here on the rock that we see down here. That's what I call an ice sponge, because I have no idea what else it is. And they're like impossible to get because they're so thin.
Swiftia poking up from behind it on the right, those red, slender red branches. I think that's another ox coral.
The Swiftia?
To the right of this, I believe.
Okay.
Video's clear.
Okay.
Lasers on.
Oh yeah.
Oh yeah.
Just
off the
peeking
back.
up behind
Yeah.
the rock.
There's also this yellow.
Is that yellow? Do you know? Is that
I don't know.
another Gorgonian?
That's
Oh, yeah.
Paramaricia for
Paramaricia
sure.
for
It's got
sure.
the classic planar
Paramurcia.
shape to the colony.
Mm-hmm.
And then with that one brittle star hanging on it, classic.
You said Paramurcia?
Yes, I did.
Okay, thank you. Or Paramurcia.
Now that's one of your aspire targets for connectivity, right? The Paramuricia plus the The brittle star?
Believe so.
That's correct, Alan. There's several species. It's really hard to tell these species apart, even in hand, and even the genetics. It's really subtle, but they're very widely distributed, a very common genus, and so yes, it is a spire target.
So would we like to add this to our collection while we're here looking at
it? Sir collects a lot.
I'll leave it to you guys. Alan, you're right. This is one of the ones that Andrea identified.
Yeah, and I saw it on the Aspire list. I don't know.
Very slight tilt down.
I think it would be smart. We haven't seen very many of them.
Yeah, we can get it.
Yeah.
I'm fine with that.
Yes, Pilot, if we could collect these two together, that would be terrific.
I would imagine we're going to have to... Are we going to jar them together? They
take difficult to take apart?
Yeah. You're either going to have to cut...
I would just jar them together.
Yeah, just jar them together because we're going to have to cut that ophiuroid out, and he's still going to have it be attached to those. There's no way we're getting him off that Gorgonian.
Yeah, that's what I figured.
Yeah. I mean, you can. It takes a little while. It might be worth snipping the tip of one of the arms.
Oh, yeah. No, we'll definitely take a separate DNA. But as far as the actual sample in the jar, I would like to keep them together in the same jar, and we'll just get 2 DNA samples from them. I think that'll work.
Not so much time.
Starboard pin
they can be separated. But I did want to say, you can clip the branch below where the coral is, where the sea star is.
Yeah, and put him in his own jar. Yeah.
Catch that.
And this is...
I'll follow you out with the mini.
It needs to be cut. You can't just snap it. So this is a cutter, not a snapper.
Hey, pilots, this is going to be really strong to the rock, so your best bet is to use your snippers and clip him below, near the base, if you can get down that deep. Thank you.
I'll just center it up.
And just to be clear, Wesley, this is going to go into one of the bio boxes, not a suction jar.
Correct.
Bio
box, yeah. I would think he's too big for a suction jar, but...
Yeah, it's just...
Yeah. This is going to be sample number 6.
And this is for connectivity study.
Oh.
Looks good.
Looks pretty good.
This is number 6.
Following you down.
Hey, John, I think I can hear you typing. Thanks.
Nicely done. That is just
awesome. Look, and we left a stem, so maybe he'll regrow.
There. All stopped.
only if he finds another brittle star, or another brittle star finds it.
If you need more light, I could bring
So
one
these
aboard.
samples that are targeted for connectivity, it's to help understand, you know, how these deep water communities are connected across distance.
Genetically.
Genetically, exactly.
Yeah. We have that constant flow of the Gulf Stream, right? That starts up in the loop current in the Gulf of Mexico and then comes around the Straits of Florida and then up through the Gulf Stream, right? But we're way further east.
So
there's probably a lot of Bohemian water coming in this way.
Yeah.
It's a little better.
I think this is where the water from the Labrador Sea gets entrained onto the Blake Plateau.
So this is a good place to be mixing genetics.
Yeah.
Right? As we're going to get probably two maybe 2 genetic populations would be my guess. We got a bunch of stuff Gulf side, and then a bunch of stuff coming up from like the Atlantic.
Nice.
All right, end collection.
Well done.
Thank you
6.
very much for that.
Forward lights down. I'm redeploying. Actually, I'll put the camera back up.
Oh, sorry, that was 6B,
6B, yep.
not 5B.
And we better think about taking some water sometime
I know.
soon.
Let's say 2. Yeah, right. About here. I need some water too.
Well, feel free.
Yeah, I'll go take a water break.
Yeah. Watch it. Let us know whenever you want the water sample.
So actually, this is Nav. A little bit of housekeeping. It looks like the Niskin 2 that we attempted to fire did fire. So I think probably 2 and 3 fired close to the same time. So we can either call that another sample at this point, or we can call them the same sample, or whatever you want to do.
Depends how far apart they were fired. We were right in the same spot when you tried to fire 2 and 3, but we don't. Do you know if 2 fired like a long time ago? We don't really know when 2 actually closed.
Correct.
Okay, I think we should toss 2 just because. Well,
yeah,
we'll talk about it. We'll grab it, but let's just not count that because I already have it not counted.
Okay.
So what's in 3 I actually counted as sample number 2.
Understood.
So the Paramir seed we just collected is number 6.
Yep.
And then the next water is going to be probably 7, unless we find something magical between now and the water.
Okay. And that would be a Niskin 4.
Yeah, and that's going to go in Niskin 4.
And we'll save the final one for up on the top of this mound.
Get another move in?
Because we already got a 1,000-meter one, too, on the way down before. Ooh, look at that.
Yay. Little comb jelly floating by. Bathosiroe, I think.
Yeah, little shrimp next to it.
Yeah.
Ooh.
What are you thinking, Copilot? 240?
Yeah, I think anything, whatever gets us to the next waypoint in that direction sounds good.
Sounds good.
Get back in on the chat, see if I'm.
There's another sea star. Look at that at 2 on that rock up there. All right. Yeah, this is quite a slope. Maybe 20, 25 degrees here. Down the slope, looking.
Yeah, it's so funny. You know, you look at this view and you think maybe there's not much living there. And then you get close and the lights are shining and all of a sudden you see that there's
tons of biota.
More things than you can
even really look at.
Yeah. We've come about halfway up this mound so far.
Yeah, I say call that water sample whenever you're ready. That's going to be number 7.
You want to do that now, Pilot?
Yeah, we're going to take that water sample when you're ready. We're about at the right depth for that.
All right, water sample firing 3, 2, 1.
So that's going to be
fired.
7W.
Yep.
Hey, Washley, just letting you know that sample just fired and it closed.
You just fired it? Is that what you said?
Yep.
Thank you.
Excellent. Thanks very much.
20:34:40 is the time. And that was at 1384. We have another sea star here.
Cirrus camera 30 degrees.
Probably Gonias asteroid. Let's see. Let me get closer. Looks pretty deflated. It's kind of skinny. Neat. He looks like he's doing pretty good on his COVID diet.
Of course. Forward lights coming on.
Let's look at that red
Nice.
tip.
A little more if you want that.
You can see his tube feet.
Really nice.
Yeah, that's cool.
Center screen.
There's also a Feria down here, it looks like, underneath
Very
him.
nice glass sponge. Might
Maybe.
be dead. It's very
Yeah, it
thin
could be.
and
Parts
fine.
of it are, right? That, like, it almost looks like burlap when they die. Hey, is this Chris?
Hey, it's Chris Ma.
I got it. I knew it was you.
Interesting.
Yeah, what do you got?
This is this is paradoxically one of the irritating things about Gonias asteroids. The more nondescript and plain-looking they are, the more likely it is that they're not something that we have described.
Oh, of course.
I mean, the really distinctive ones just come right out at you, but it's always these kind of plain ones that are irritatingly difficult to pin on. This doesn't look like either Ceramaster or it might be Mediaster.
Mediaster?
Layback is the distance between ship and...
Yeah, Mediaster. I can put that in the chat. But it doesn't have granules on the... Okay.
Yeah.
We're at...
I don't think we can put the ship back.
Yeah. Are you in a position to collect? I know you guys just did your water sample.
Stars are pretty quick.
Can you?
Yeah.
Yeah. Can we collect this, pilots, or is that? We can try, Chris.
I mean, I understand, obviously.
That could fit in the suction sample.
If we can, that'd be great. Thank you.
So that's going to be sample number 8, if we can get them.
Just below 10, it might fit, yeah. That's the last one we collected.
Do you think you just
size.
undescribed species, likely?
Just under
Is that
10
what
centimeters.
you think?
Yeah.
Okay.
Yeah.
We had to put some extra thrust towards
There's
the end to get it through, but...
probably something it's feeding on underneath it, too. I mean, there's a whole bunch of like encrusting stuff on the rock there.
Do you think the suction hose is the
easiest? If you have to move on, I have to keep the timetable. That's fine.
It's fine. We don't have very far to go.
Yeah.
Like it's a short dive.
Assuming
like a short distance for us to
flexible, right?
cover.
Yeah.
So we've really been poking around a lot.
The last one we did,
Do you
I can't remember
think
the
the
pattern.
suction hose is the best way to get him?
That size and it went.
Yeah, it might be too big for the suction hose.
So this thing is about.
Yeah, it's definitely like...
So the lasers are what, 4 inches
10.
across, right?
Yeah, 10
So
centimeters.
it's actually not that big.
It's not, but the suction hose is just too
it'll
small.
It'll come off pretty easily, I think.
Okay. You think so that we can grab him with the claw?
Yeah, that'll work too.
Okay. Oh, yeah, go for it. Sure, that works. Okay. Perfect. Perfect.
All right, going to canister 1. Yes.
Okay, I've got eyes on vehicles.
Not
yeah,
live.
I think so. I haven't seen it move at all.
And as long as I've got you guys on the phone,
Yeah.
the one that you guys saw earlier, the white one, that was a sort of a familiar predator of corals, and it was actually feeding on. I'm not sure if it was a primnoid or something.
Mm-hmm. Parameciid,
you can
I
see
thought.
the vestiges of the skeleton in the back. But that was a Cerciaster americanus, which is, thanks to Okeanos, now one of the better known predators of octocorals down there, in addition to Evoplasoma and Typisteria.
What was the name of that guy? Can you repeat it, spell it a little bit, or put it in the chat for me?
Sure.
And
I actually put it in the log.
Okay.
But it's Cerciaster.
Oh, I see it.
Spelled
Okay.
like goddess of magic, like Circe, Circeaster. This one is a goniasterid that looks vaguely like Mediaster, but it doesn't quite fit.
You got an Auto XY?
Okay.
Fit the one that's supposed to be in this area, so.
Great.
Release it down a little more. Yeah, that's where you want it. Hydraulics are up. Canister 1.
I forgot to note the genus of this.
Of this one?
So.
Your fog lights are still on?
Close to Mediaster is the closest thing I could come up with.
And Mediaster is M-E...
yeah, like Mediaster.
Who's the extra
Good.
lights?
Mediaster.
Lights coming off.
Yes. The name literally translates to medium star, so...
That's pretty easy to remember.
Off. Good grab.
Yeah.
All right, so he's going into suction. Great.
All right. Retracting drawer. Ready for that?
205. This is
Tracking
sample
drawer now.
number 8.
Not until we get it in.
Oh, yeah.
Yeah,
Copy.
it's attempted.
Don't cross the beam.
Yeah, I think that's right about where your last perch was.
Yeah.
Looks super steady.
Looks like a good spot.
You got the light on the jar?
It was. Let me double check. Suction light is on. You can turn it all the way up if it's not in color.
It's in color. It just looks dark.
Oh, that's all the light we have?
Okay.
Sunny Mini Zeus. I don't know if you want your PTZ at all. It's pretty blocked actually. Yeah, ready? Nailing suction now. You're inhaling.
Looks like
Canister
maybe.
1. Increasing. Do you just want to put it in a box, or do you want me to increase 1,000? Increasing.
But if it
That
turns,
looks
it could get
pretty stuck.
stuck.
Do you want to decrease full?
I can see him at the end of the tube.
Mm-hmm.
Yeah, we can too. I think they're either going to try and suck them in or turn around and blow them out into a bio box. Not sure.
3, 2, 1.
Sea Star saying no thank you.
How long is the suction hose?
How long is what, John?
The suction hose. How far out can they stretch that thing?
Oh, pretty far.
About 6 feet.
I'm not sure. 6 feet. There you go. 6 feet from the ROV.
It's fully disabled. Do you want to keep a little bit going? Something in port enter.
Starboard enter.
Yeah.
Okay.
Get your views you want.
Pins extended on starboard firebox. And then just straighten the hose out a little bit before I re-extend.
Get your lights set.
That looks pretty good. I
Yeah,
think.
looks good.
Yeah, let me know when you're ready to extend. Extending now.
Will do.
So what about... No, you want to go a little more? Do you want to extend it a little more before you... Alright, so that's fully extended.
Just make sure you don't tilt it down before. Yeah, there you go. Yeah, it's the hard point.
You know, we've got so many things stuck in these corrugated tubes. It's kind of tricky sometimes. Sometimes you have to wait till you come up and take the tube apart.
Just put the whole thing in there.
Yeah.
That might not come off. So get the sample first, and then we can deal with it.
Just... Yep.
Get in there.
I'm watching this and just, I hope that's not permanent damage.
Well, it is for
Talking to the masters. Yeah.
Could spring it back.
Nah, let's get the sample, and then we'll deal with the nozzle.
I think if you try to get it a little deeper and just kind of tap it on the edge of the box.
Use some blowing action too.
Yep. Keep doing that.
Oop, oop.
It's almost there. Yeah. It's gonna
I think
go.
so. There
It's
you
going.
go.
There it is.
It
Well
is. Thank you.
done. Starboard inner.
I think
Was really impressive.
I say we either.
I think we could put it in the port rock box, maybe. So, watch your
If
camera.
you guys want to tuck it in the
Hit the
bio box,
camera.
that'd probably be okay.
I'm worried that's gonna sever.
Okay, good. It's been gotten.
Yes, it's in the box.
Okay, lovely. My kudos to the pilots yet
What if
again
we-
for a
Right.
remarkable collection job. Thank
Try
you.
to snip it and put it in a bio box or rock box.
And they will
we
send
don't want it to
Chris
dangle
the
like
bill
that.
for the new suction hose.
Right.
That's right. Be on the lookout.
Yeah. Okay.
Sure.
Well, I was thinking you'd use it to open it, but
Well, that's definitely something you don't see every day.
I think you
Yeah.
could put it in the starboard and then just trim it. Just starboard rock box. What do you think? Just wing in.
Yeah. Stand by. Sample box.
So we have about 40 minutes left. I say we make it to the top of this mound.
Yeah,
Make
I
that
would
the
like to
priority.
see that. Yeah, move
So.
kind of fast
Thank
until we
you.
get up
I'll
there.
talk to you guys
Yeah.
later.
Yeah, no
Thanks again.
worries. No worries, Chris. Thanks for calling in.
It's pretty elastic. It might just.
Sorry about the
pull
tube.
Oh, jeez. They'll fix it. It's no big deal. What's up, John?
Alan and Stephanie.
Yeah.
I would suggest going to the top and
Yeah.
spending the last 45 minutes up there.
Yeah.
Then what
Quite
if you just
different.
Yeah.
Or it
try
might
to
be the
count down
same.
and snip?
Yeah.
Yeah, it might be. That's what I said. We're going to make our way up to we're going to shoot for the top of this mound now, and then we'll look around up there before we leave. I'd like to get up there before we end the dive. We have about 45 minutes left. There you go.
All right. Do you want me to retract now? Retracting drawer slow.
Yeah, I think so. If you can grab it.
Retracting pin. I can extend it back out. Retracted this pin. Do you want to re-extend the drawer a little bit? That's there. Do you want to get the lid open?
What's your impression of the sponges that we've seen today? You know, a lot of different kinds, but most of them pretty small or not so much?
We had a few large macro sponges. Let's see, we have a couple
And now,
ficalias.
do you want me to extend it?
And let me look.
Extending drawer.
Pretty diverse. A lot of them I don't know their names, so I don't know how helpful that is.
Some
But
Phoreas,
How's that?
maybe Euretid, definitely that Bolosomen.
the one we collected, the
Yes,
big, yeah.
I feel like we've seen quite a lot of small, like when we've zoomed in on the rocks, seen
Oh,
quite
yeah,
a lot
definitely.
of small, different,
Unidentifiables.
definitely unidentifiable from. I'm gonna save that.
And then go for lid.
Go. Take a look at the top.
Nice. All right, wing outboard. Retracting.
All right, so we're going to store our collection skid.
Such a landscape.
Valve packs off.
Yeah, I'll
Yeah,
make
it's a
that
rock boulder field, pile of rocks.
waypoint 2 active
Yep.
here.
And now we will be making it up probably about another 50 meters to get to the top of this thing and spend the remainder of the dive up there, checking up the top.
Yeah, and I say we kind of just don't really look at anything too closely and let us at something new that we haven't seen yet.
You
Maybe
mean
some snap
between
zooms.
here and there?
Yeah. Like between just some snap zooms, maybe.
Right,
Like
should we get a move in then? Sounds like they want to.
there's a couple of yellow sponges.
240, I'm thinking 20 meters.
Yeah, sounds
Okay.
great.
It's hard to
Bridge nav.
actually, if the snap zoom is possible.
Yeah.
Could we do another ship move, please?
You want to snap zoom that yellow
Range
sponge?
20 meters. Bearing
Can we get a good
240
zoom on that yellow
degrees,
sponge on
speed
your way
0.2
past?
knots.
One dead center. They should be the same species.
Good copy.
Looks like a Leiopathes maybe too. Oh, that's pretty.
Oh, that's nice.
A yellow hexactinellid.
Yeah, I think that might be Hertwiegia. It's sort of wide open.
Yeah, I think it's hurt Regia.
Yeah?
The Chris is back, so you let us know.
Yep.
Okay. That's pretty cool. There's a worm inside.
Yeah,
a couple
really
of worms
nice.
inside.
So that's the first time we've actually seen that, even though we'd seen some other larger yellow sponges that made us think that we might be looking at Hertwiegia. When we zoomed in, it wasn't. But this is, most glass sponges are nice, bright white colors, and this one is it's really distinctive yellow.
Okay, we're good whenever you guys are.
Thank you for that zoom.
Christina agrees, Hertwiegia.
Nice. Looks like this pile of rocks is a little more broken up here. They're not quite as large. More of like a 1 meter size, half-meter size boulders.
Yeah. Let's see. Rotating camera up a hair. That's 35 degrees there.
Again, we got Stichopathes. Sea Whip at like 8 o'clock. Maybe another Anipetharian at 6.
Mm-hmm.
Yeah.
Affirmative. Well, actually, we got
What
about
is the minimum depth of this?
1331, John. Sorry, Nav, go ahead.
It's okay. We got another 200 meters to the top.
Okay.
So we're going to just probably keep it moving unless you say otherwise.
Yeah. That's what we're shooting
Sounds
for.
great.
I'm going to get up there before the dive's over. We need our last water sample at the top there, too.
Understood.
Yeah, it's the same similar species we were seeing a little deeper. These yellow Stichopathes look like the most common. And that Paramuricid Gorgonian here, there's 3 at 9 o'clock. Each with their own ophiuroid in it. There's another Bathypathes at about 8, that feather-like one. And some more Leiopathes.
The geology of this is really weird. We have to get a geologist to
right?
tell us what's going on. the multi-beam, just shows a flat plateau. You have this mound, maybe a half mile away, another mound that are perfectly conical, very typical coral mounds, and clearly this is not a coral mound. How did this thing
get here? I have no idea. Before you speed off at 9 o'clock, I've got another rock pen. It just went off screen. It is coming into screen right now. We just get a good video of that.
Sure. Same thing. Okay.
So this looks similar to the one we got earlier, which is this rare morph that grows on rocks.
Yeah, just a bit larger.
Look at purple gorgonian behind it too. That's neat. Sweet. I would guess the same species, but it looks very similar.
It does look very similar.
Okay. Okay, we're good, thanks. There's some cool purple gorgonian behind it. Another black coral. See the black stalk?
Yep.
With the red flesh.
Clear.
It's probably, that's a Stichopathes. There's maybe a stalk crinoid.
Yep, I'm moving.
Yeah, still got a ways to go. Creeping up to the top of this deep mound.
It's like a random pile of rocks in the middle of the ocean. Let's see, 9 o'clock, I see a possible corallimorph right at the edge of screen. Get a snap zoom on that. Nope, down further. Yeah, that's like an orange. It may not be corallimorph, it might be a, yeah, corallimorph. You can see the foot around the outside.
Foot.
Cool. Thank you.
See that on the section right?
Yeah.
Copy.
I dimmed it down.
Really large. About 10 centimeters across.
Yeah.
It's on now? Is that what you said?
I had to dim it all the way down. It came up and it's on
So
now.
now Scott thinks that was maybe a
seem like
Hormathid anemone.
Oh, Hormathid, yeah.
You can see
Oh, there you go. Yeah.
That's Max again.
Huh.
Isn't Hormathia the fly trap?
Yeah, the flytrap is in that family, so I mean, I don't know
Gotcha.
for sure that it's in that family, but I do believe that is a
so it's
true
harder
anemone
to,
actinium area and not a corallimorph and something close
Understood.
to that.
Thank you, Scott.
So Kimberly Galvez chimed in about the geology of this place.
Well, that's right. Since we're on the edge of the Blake Plateau, this looks to be the exposed portions of the outcrop from the edge of the plateau. Could be broken up chunks of limestone that have since been encrusted with ferromanganese. Thanks, Kimberly.
Looks like it might be a hair steeper, but
These Lyopathes, these orange
I don't see any walls or anything coming up.
black corals in the front of the screen.
Yeah, that's not real. I've been watching that on the lower one. I don't know what it is, but
As far as I know,
it's gone now.
Leopathes are the so far oldest known corals that have been aged using geochemical techniques. More than 4,000 years old, some colonies south of Hawaii.
4,000 years. That's
Did you
incredible.
change the frequency on the bottom one? Yeah, you can't see it interfering with the top.
I think still now the record is deep sea glass sponge. It was dated to 11,000 years in age. Yeah.
Oh my goodness.
Wow, Alan, I did not know that, or I wouldn't have said that. So how do they age it?
I think that was isotopes on, because it's one of those ones with a giant single axonal spicule that's many meters long, and then it's laid down in layers, and I think the original layers were dated.
Looks like your wire cam's
Looks like
up
a
today.
bamboo coral here at ooh, there's a cool sea star.
This morning. Again,
Can you guys swing
Copy.
to the right behind the coral there? There's a
The
poofy
actual
sea
monitor
star at
of the
noon.
camera.
He looks different than the other ones we were seeing.
I don't know if you copied pilots. That move is complete.
Copy that.
Good with that, co-pilot?
Yeah, I probably still have another at least 10 meters of swing.
Yeah. Okay.
Is that one of those cushion stars? That's kind of squishy. Neat.
Confirmed.
Wow. Looks
Awesome.
very soft.
Mm-hmm. Like that pillow foam in your sofa. Squish it.
Said you ready for that move?
Hey, Chris, how are you?
I have another 5 meters.
What do you got?
Hey, so that is a pterasterid. It's probably
Good with that.
pteraster, I think. So these are slime stars.
Mm-hmm.
If I collected one of those, or if you guys had collected one of those, chances are I haven't actually tested this yet with the deep sea ones in the Atlantic. But if we pick those up, they emit mucus as a defense mechanism.
Oh,
and
yeah.
So that would be fun, I think, for you guys, if that was one of those things that we were going to test. You would probably get a box full of slime to work with
fun.
when they come up.
Floating fish.
It's not.
It's not.
That's the fun part of testing these things in the deep sea. But I remember both pteraster and master can create a fair amount of slime when they're being harassed.
Are they
the animals themselves?
Go ahead.
I'm sorry.
I was going to ask if the animals
well, I was
actually
gonna say, they're really weird on top of. Yeah, they are squishy, but they're also kind of crunchy because they have spinelets in the. They have what's called a supradorsal membrane, which is kind of a soft tent over their actual body surface, and it's held up by sort of calcareous, spiny tufts, and so basically, even though they're very fleshy, they still have all of these sharp, small bits in them, and so handling them is. I mean, like with pteraster, it's not such a big deal, but with hymenaster, it's a challenge because hymenaster is almost completely gelatinous. So that one in particular probably still had some skeleton to it. But like I said, their architecture, the body shape, is really strange because like they have they have a false layer over the top, below, and then the actual surface of the animal is below this circus tent layer, which is on top. So they're very unusual, and there's a whole bunch of stuff associated with them that is interesting. But
awesome.
I'll let you get back to. It looks like you have another rock pen.
Rock
thing.
pen,
So-
yeah, and another Antipatharean in front of it.
So that's like
Have
another.
we seen this
Looks
this
like another
black coral?
interesting bunch of stuff going on. So I'll
Yeah.
talk to you guys later. Hopefully, another starfish will come up before the end.
Okay.
Thanks.
We
Bye.
have not seen this black coral yet. This looks like Tenacitipathes, sort of, that we have on the like in the shallower mesophotic area, but the polyps are way different. But they have that same growth pattern where it looks almost like you ripped a piece of a pine tree.
What's it at?
Like a Christmas tree growth.
Yeah.
Like a pine tree branch. But I don't know which one this is besides Antipatharia.
It's like
That's
700%.
as good as I can get it.
This is a great view of it.
And then another rock pen.
Yeah. Calibalemnon.
Yep. Hopefully.
Calibalemnon franci.
Franci.
Is that what is on there?
It's not?
Ah,
Oh, I don't know. It looks like something overgrown like on the main stalk there.
take a-
Maybe a parasite. It's overgrown a parasite or something. It looks like and a Bathopathes behind it. Oh, I saw Bathopathes too, right?
Mm-hmm.
Copy that.
Batho or Bathy?
It's Bathy.
Yeah, Bathopathes.
Again, these rocks look like they're getting smaller as we get shallower.
We're up about 1,367 meters.
We have about, 15, 20 minutes left?
Got another yellow Paramaricia.
Looks like white sponge up here, maybe. And what was the name of this one with the stalk? Do you remember?
Oh, that's a Metallogorgia.
Metallogorgia. We've got a white. Look at this white sponge back there. Can we get a snap zoom on that weird sponge at center screen? Looks like a funky mushroom.
That is really cool.
What is that? That's weird.
No, I think that's
Hexactinellid?
I do think that's another hexactinellid.
Yeah, it looks like it's dying though. It's got that burlap texture to it. I don't have a clue what that is.
It's not conolasma, is it? Oh no, it looks like a tall note australis, says Chris.
Looks like it's definitely.
When you go
pulling
light
the
on this, can you look under the
sediment
rock to
inside
the right after you
and
finish with
blowing
the
it out.
sponge?
Under the rock to the right.
Now, after you finish with the sponge, you'll have to pull the camera back out. It's much closer to where we are.
So go full
Something,
wide.
reddish.
And then there's something reddish.
I'm wondering if it'd be Conorite. I don't know.
The single stock here at
Oh, no.
at
Straight
6
down.
o'clock, or the one on top of
Below
it?
that black, yeah.
The single stock. Can we look at that
It's not
single
what I
stock?
thought. It might not be important after all.
Okay.
Lycon,
Well, they're all important.
extra lights.
Antipatharean,
Yeah.
maybe? Yeah, Antipatharean of some kind.
Fog lights on.
Looks like maybe a cut off Stichopathes, but its polyps are huge. So, yeah, we're not sure what that is. I don't know.
It's an odd one.
Yeah. Okay, that's good. Thanks, guys.
Yeah, stick patties. But that's not at all what I thought. I was kind of looking for an Acanella bamboo. Not that. Very cool. Thanks.
Here's another Paramyrceid, maybe. It's at 9. And then we have 3 Stichopathes or whip corals right in the front. And we only have about 15 minutes left. Ooh, another Faria possibly over here at 3. And then this one I cannot remember the name of.
Metallogorgia.
Thank you.
We're gonna have to, so we got about
So probably close to being settled.
30 meters of relief to go.
Do you want another move to go?
And I think about 15 minutes.
Mm-hmm.
I think that's another Hertwigia.
That was a big fish. Got a big fish at dead center.
Can I use your blue view pilot?
That looks similar to the one we saw earlier, but this one's skin doesn't look as... Oh, no, it is different, isn't it?
It's different, yeah.
Laser's clear.
I think, anyway. Last one was more eelish, right?
Mm-hmm.
Yep.
This one looks more
This one looks
morid.
to me like a hake.
Hake.
Or something.
Yeah.
Again, I'm going way out on a limb. I'm not good on the fish.
One of these deep hakes.
Cool.
Tracking iris, tracking focus.
There's a little piece of Faria here at 8. Or 10, sorry.
Keep moving up.
This could be a... Let's see. Could this be a... No.
I don't have a clue. What do we think?
No, not that. No, it's not that. Because the tail's all wrong. Trying to look through pictures. Not the smartest way to do it, but...
He's a giant eye. And a big nostril, too. You can see the lateral line above the pectoral fin. That's how they sense changes in water pressure next to them.
Mike Vecchione agrees that looks like a blue hake.
Nice. Oop, and then a little guy next to him.
Oh, good.
That one looks more Lamanema-ish. It's a morid codling. Hey, somebody chime in?
Hey, this is Mike Vecchione
Hey,
here.
Mike.
Did somebody chime in?
Yeah, Mike Vecchione here.
Go right ahead.
Yeah, that's the Entomora rostrata, the blue hake.
Blue hake.
Really cool.
Thank you.
Yeah, that's the Entomora rostrata, the blue hake. They're really common farther north at this depth.
Awesome.
They're really common farther north.
Yeah, I probably haven't felt the last
Can we get a quick zoom on the Metallogorgia as we go by? It's
Like,
like it still has lower branches.
yeah, can we zoom in on this Gorgonia on that center screen? I think it's growing off that one. I think that bottom one is dead. Yeah, and it's growing off this. And again, we've got this little pink hermit. 2 of them.
Very
But
cool.
looks like it's growing off that.
I've never seen. Yep, I've never seen a Metallogorgia growing off another one like this. Awesome. And there's those dang hermit crabs again. All right, moving on up. Thanks.
Yep.
Question is it growing off of, or is that just a still alive of the same species?
Wow, that really is.
I wonder if is the other one a Metallogorgia too? Or is it a different species? It seems to be similar, but it's not quite the same growth. See the netting of the main branches looks different on the dead one.
I think it is, Stephanie.
You think it's the same?
It's got hydroid growth all over it.
Okay.
It's definitely a Chrysogorgiidae, which is the family that Metallogorgia belongs
Winds
to.
off bottom.
Called the Chrysogorgia. But that's what it looked like. You had the 2 main bifurcation point branch.
Awesome. Thank you.
One
I'm
more.
really curious about all
Do
these
it now,
hermit
yeah.
crabs, though. I said at the beginning of the day, I've never seen one crawling up on these tall axes like this.
I don't
and
know if
now
it'll
we're seeing all kinds
we'll
of
probably be moving.
I wonder
we're
if it's
setting up at
seasonal.
7:30.
Like
Sure,
maybe
too.
they're coming up, like it's mating season or something. And I don't know.
Okay.
Just wild guesses here. That's all I got today.
special mystery mound hermit crab.
Mm-hmm.
And we still have we heard from anybody about what created this mound or what the idea is there?
Kim's had a commentary about it. She was talking about it being a buildup of rocks that were like left over from the. I don't know. I have to find her thing. Kim.
No, you have a little more. I'm looking
Let's
at
see.
you.
See if I can go find her comment.
Chunks of limestone blocks that
You still
could
have
be
a
the
nice
result
looper
of
here.
shedding from
Seam
the
in
outcrop
tightening.
and build up
See that?
over time.
So,
But why the mound?
Well, yeah, well, why the mound exactly?
I'll let you know if.
Ooh, that's a big stick of pathies. Oh, we got a cool fish. Went down at 6. Little pink guy. He's in your list.
Yeah, I'm sure he is.
That is a Chonax.
Chonax, yes.
He looks like a balloon. He is a very grumpy fish. He looks very angry.
Oh, or is this a
Stephanie.
goosefish?
Yeah, John, what's up?
If you look at your multi-beam, you got a long way to go to the top.
Yeah.
If there is hard coral, it's going to be on the tippy top, not
Yeah.
this rope.
This is a gaper.
Gaper.
Chonacops roseus, it looks like.
Watch the lead, Nav.
Yeah.
It
Go
does,
ahead.
I don't think we're going to make...
Don't think we're going to make it?
Yeah.
Okay.
We're still about 100 meters.
100 meters?
Or more away. And we have 8 minutes left.
Okay. So let's pick up, speed up, go as fast as you can. And we'll get as far shallow as we can get.
The ship,
No more looking.
ship's been constantly moving, and so is Sirius, so we can't.
We're kind of going as fast as we can.
Yeah.
Understood.
The looking is up to you.
No looking. Just go. Yeah. Everybody close your eyes. We'll be at the top in a minute. So Chonax, is that what you had? Is that what it, was that right?
I thought it was Chonacops, but that's what
You can get me the name later. That's fine. Pink.
Yeah.
Yeah, we've been a constant move, watch lead, so we haven't stopped hardly
Yeah.
at all in the last.
The rosy coffin fish.
I figured I just timed it bad. We just,
No.
we just need to barrel through here. Oh, that we may not make it to the top. So we're going as fast as we can. Yeah, we've been moving nonstop since for a while now. So we're just going as fast as possible.
Basically, since the last collection,
We were trying to
constantly
get to the
moving.
top of this, but I don't think we're going to make it. We only have about 8 minutes until we have to come up. Here's a cool yellow one. Yeah, I'm not allowed to look at it though. Gotta go quick. Don't look. Yeah. Yeah. That looks like Leiopathes to me. It's a little yellow Faria, possibly. Some white Faria here at 6.
Here's where we're gonna start seeing the Medusae.
So you can collect more?
Can we?
No. Yeah, the boxes are full, and it's like, yeah. So I can see up this hill, and I see some gorgonians, but I don't see any. If there was Lophelia, it'd be white. You'd be able to see it from here far away. Ooh, here's a tube, a white tube, hexactinellid.
Oh, so another euplectellid maybe?
Oh, that's funky looking.
Oh no, it's not. Doesn't have a sieve plate on top. Just a thin.
Just a tube.
Tube.
Euplectellid and There's another one too, isn't there? Heterotella? The wedding sponge.
Oh, Euplectella,
Euplectella.
and Regadrella.
Regadrella.
But yes, but it's not one of those because it didn't have a...
He asked.
Oh, sorry.
what the-
I'm sorry. I was a little
Roger Bridge.
busy in the chat room. Sorry.
5 minutes left in the dive.
Copy. Is that at 5 start setting up?
That is a fish. Little scorpion fish,
One
maybe?
big one?
No, it's one, I think. No, I have no. It's some kind of hake or.
With some. Is that me and fog lights? Copy.
Oh, neat. Look at his dorsal
That's
fins
the Chinese code word.
moving around. Look how long he's got these tendrils, too, underneath. The feelers, maybe?
It's a really interesting looking fish.
He's also got chew on his nose coming out on his face. 2 feelers. Come on, get out from under that rock. I want to watch you swim. And you just swing a little bit to the back. You just passed over a coral. I wanted to get a quick look at that guy, that white guy right there. Okay, Gorgonian.
Look on the bottom. There's a bunch of rubble.
Bunch of rubble on
Try
the
to zoom
yeah.
in on that.
That looks like it could be Lophelia rubble here that it's attached to.
Corallium again.
Yeah.
There's some sort of rubble all over the.
Yeah, there is. Can we get zooms on this rubble here? That might be Cara rubble. Yeah, it is hard to tell. Let's see. I'm not sure what it is. It could be a Cara rubble, John. I don't. Yeah, right here.
There in the lower right.
Yeah.
Actually.
I don't think it's Lophelia, though. Yeah, okay, guys, okay. Stop looking, we gotta go.
Science, if you look at string 3 or look at the Sirius view, you'll see that the Sirius hasn't stopped. Sirius is still moving. So we're not stopping.
Okay.
We'll just remember to...
We're trying to get as shallow as possible, and then right before we come up, we want to snap that last water sample. Some different black corals, it looks like. It looks like we're losing our rocks, too. Getting more sedimenty.
Yeah, it's interesting. That
There might be a little white coral there. Yeah, a little Gorgonian. Snail.
nice little gastropod peeking in there.
Hey, pilot, how you doing? What's the thing go time?
Right now.
Okay.
All right. Watch, that's the end of this dive. We're going to start setting up for recovery.
Okay, can you
just snap the water?
Yep, give us a water sample.
We
we
can indeed.
Thank you.
Thank
you want it
very
right
much.
at you want it right at 1350? We're at 1352. Closer to the bottom or up at that depth? Watch leads. Watch leads. You want the Niskin right at 1350 or 1352 near the bottom here?
Either way, it doesn't matter.
Okay. Sean handed off that he wanted it. They wanted it at whatever. So
That's what they said. Yeah.
Yeah, this is fine.
Okay.
I think if you want to go ahead and fire it.
You guys ready now?
I'm ready.
Dive 5, sample 0. That should be 9. 9W.
Release. Do a reset. And fire. Niskin closed.
Got it?
Yep.
Thank you.
Thank you very much.
Drop in the target.
Okay.
Logged.
I'll do your swing arms here while I get you. Lower swing arms. Outboard. And stow. Careful. Slow.
Dive concluded. Well, that was awesome.
Yeah, it was. So sorry, John, we didn't make it all the way to the top. It was totally my fault.
Swing arms stowed. Disable. Upper swing
We were taking too long.
Down.
There was a lot to see.
So just to recap, if you're just joining us at the end of our dive today, this was actually our second dive today because our first dive, we had a mechanical failure on the RO base, so we had to recover it and then relaunch in the same spot. Let's see. We are 174 miles from Jacksonville along the Blake Plateau, along the
eastern edge?
Yeah, the eastern slope. And we traveled from about 1432 meters at the base of this mound up to, we got up to 1350?
Yeah.
We took 9 samples. 4 of them are water.
Okay.
Right?
Yes.
Rising all the way. You want me to wrist out
let's see. The total rise for this mound was about 101 meters.
Oh, okay.
We didn't quite make it to the top, though. 18-degree slope. The length of this feature is 620 meters. And then we had planned a 545-meter dive, but I'm not exactly sure how far we actually made it. That would be post-dive data. I'd have to find out.
And we were curious if this was going to be a biogenic mound, but it turns out it was.
You want me to come up 10 meters from
your
cap?
There may have been some coral rubble there at the end. So there might be some coral at the top of it.
On my way up now.
And unfortunately, I'm a slacker, and we didn't get it there.
We had a lot to look at.
Yeah. We are going to have a post-dive call and a pre-dive for tomorrow call after this. I believe as soon as we're done broadcasting. So if you want to be involved in tomorrow's dive, go ahead and stay on the line or call in. We are going to one of the deep dives on the Blake Spur tomorrow, and we'll be on the Blake Spur for the next couple of days. And then we'll make our way back across the Blake Plateau. So go ahead and stay on the line. We'll be with you guys shortly while they do recovery.
Right when you're done? Okay, great.
And I think we're going to go eat dinner as quickly as possible before we have to process all these samples. Okay, so thanks for diving with us today. It was really fun. We hope you join us again tomorrow.
I stopped Delta 10. You want
anything else, Alan?
No, not a whole lot. Just thanks a lot.
Yeah, thanks, and thanks for chiming in and being involved and helping expand.
Tomorrow we're going below 4,000 meters,
yes,
so it's going to be
one of our deepers.
More exciting.
Yeah, one of our deeper dives. So just hang on the line. We'll be with you shortly.
Gotta go right. So I'm going to spin right. Should eat those out just fine. Looking good. A little bit further around to go. Checking to put auto heading back in once I'm lined up, or are you good like this? Okay. Okay. Copy. Sounds good. Hauling in. That's probably a little less than 30. Yeah, 23, 24, increasing. 26, 27. One more bump. Yeah, one puny rock sample, I think, is all I saw. There's 30.
Go ahead, Bridge.
Okay.
Yeah, as long as the center of rotation is at the A-frame, we're good with that. Copy.
Where are they headed? What heading?
Yeah,
Starboard.
to starboard, yeah,
Okay.
just 5 degrees to starboard.
Got it. Looks great.
So,
oh, winch is at 32, 33 now. Sped up a little on me. Slowing down. Another notch. Let that stabilize. See what it averages out at. 29.30, so that's better. We're on our way up. I know for a fact they didn't see it physically go off, so they don't have a time. So, but I know that we zoomed in with Sirius and confirmed that it did not go off originally when we first fired it. So, you know, it's
Go
pretty
ahead,
much
Bridge.
at some point during the dive it went off. We don't know.
Copy.
It would be
Heading change complete.
We could try to narrow it down
Also,
looking at video
we just
feeds
left bottom
and see if we can like catch, you know, try to work into where it was based on, you know, their recordings. But that would be you have to check in with the video team. That might be a lot of manual labor looking for that. So yeah, well, I mean, if it's worth it, then it's just up to them to make that call, if they're capable or whatever. They can usually zoom pretty fast in the video and like at least see the change, and then like have a small spot to run the video slow through. So they might be able to do something like that. Art's not on a headset, but I could ask him. What do you think, video? Is that possible?
I didn't hear what you were saying.
They're trying to catch when the Niskin went off.
Yes, we definitely had that recorded. It was recording the whole time.
Okay.
So we can back it up and search for it.
Yeah, do you mind? They're curious what time that was. Yeah, it looks like they're going to work on that for you. Watch lead.
Okay, pilot and co-pilot, just to update you on the situation. The current subsea, it seems like at around 800. The westerly current starts picking up and it increases all the way up to the surface. But it's not that significant. At the surface, it's, well, actually, it's about 1 knot.
It's interesting
Mostly
the way
west.
we're laying back, huh? Look at the layback's there, but the vehicles are streaming that way. It's kind of odd. Just like topside current.
I mean,
yeah,
yeah.
That keeps going out. Like,
yeah, as long as we don't get warnings, I think it updates.
Yeah, those yellows will line up. Yeah. I think it's just current and stuff. Ships. Oh no, that's the old move.
Oh,
that's right.
sorry.
I have no idea what the current is down there. I just know that at 1,000 there's very little current, and then at around 800 you get a westerly current increasing all the way to the surface, and then at around 700 there's a northerly current, but it's maybe like less than half a knot all the way up to 200 meters, and then it dies down. So if we want to start
streaming, looks kind of cruddy. If we want to start coming ahead,
Okay.
over ground even, just a half knot.
Okay.
I don't know. We're already doing 8/10, so I think if we start backing down.
Okay, yeah, we can definitely like start coming ahead of the ground, and then we'll see. I think Chris is having dinner right now.
Okay. I think just like a position move, half knot straight ahead for a few hundred meters just to get us streamed nicely.
All right.
Is all we're looking for.
Yep.
They might even be able to just dial in a half knot over ground without a end position, if you will.
Yep. Bridge, this is Nav. Is it okay if we can request a move? Just pretty much go straight about 0.5 over ground. You can put maybe like a 200 meter. We just want to make sure the vehicles are streaming correctly before we proceed. Yep. Yeah. Thank you. Bridge, this is Nav. And then please let me know when we get to, before we get to the end of the move, we probably will want to prolong it. So we don't want to come up to a stop at the end. So maybe make it 400 meters just to make sure. Thank you. All right, 400 meters. Where's that?
How fast are you going?
I asked for
on this
0.5.
Sorry, not. I was.
Oh, okay.
Sorry.
Okay, so we should get to the end of that move around the time the RV actually reaches 50 meters. So I think we would be okay there.
Copy that.
And hopefully streaming improves.
Yeah, we'll keep an eye on it. Right now we're about 65 degrees to starboard. It is odd how we're streaming though, with the layback.
Yeah, I mean, at around 800, you see a northerly current picking up. So I don't know what's down there.
Copy that. Yeah, we should be coming up on that here shortly.
Do you want to put a little bit of starboard way, or you don't think it's necessary?
Not quite yet.
All right.
I think Sirio should feel it first, and then start pulling us. And if we're starting to get so if we have an eastern component once we hit 1,000, a little bit. Or is it all west?
No, like it's maybe eastern about from 850 to 800. Just a tiny little bump.
Gotcha. All right, yeah, I'll put a little bit on.